Hello, here is Pike, Chair of APRC. I wish to extend a warm welcome to all attendees to this event. APRC is proud to host the second IBRO Global Neuroscience Webinar Horizons with the theme on Translational Neuroscience and Novel Therapeutics for Neurodegenerative Diseases in the Asia-Pacific region. We are proud to co-host this event with FAUS. Neurodegenerative diseases is a national, regional, and global burden. We see there's an urgent need to escalate the research program to understand and to combat neurodegenerative diseases. This webinar series provides a perfect platform for global networking and to raise awareness of neurodegenerative diseases. Today, we are grateful for having 6 speakers from across Asia-Pacific region to share their views on the advancement of translational neuroscience, new evidence of novel therapeutics for neurodegenerative diseases, and finally, where are we heading in future research? For that, without further ado, I would like to pass the floor to my co-host of the day, Dr. Lin Hui. Stay tuned. Thank you.
Hi, thank you, Betsy. That's a really good introduction. All right, so I will start introducing our first speaker. Our first speaker is Professor Cliff Abraham. He's the co-director of the Brain Research New Zealand, as well as the founder of the Brain Health Research Center at the University of Otago. Prof. Abraham received a PhD in neuroscience from the University of Florida. He then undertook postdoctoral research at the University of Otago and the University of Gothenburg before taking up a lectureship position in University of Otago. Prof. Abraham has attracted and managed over 60 million in research grants. His research is focused on the neural mechanism of learning and memory, especially the phenomena on long-term potential, long-term depression, and metaplasticity. Currently, he's a director of a large program of research investigating biomarkers and therapeutic agents of Alzheimer's disease. So for today, Prof. Abraham will talk on addressing aging-related neurological disorder in New Zealand. All right, Prof. Cliff, the floor is yours.
Okay. Right, we'll get started in a moment. Well, hello everyone, and thank you very much to Pixie and the APRC committee for inviting me to give you a little bit about what's going on in New Zealand in terms of translational neuroscience with particular focus on neurodegenerative disease. So I can only give you snapshots of some projects that are going on, but hopefully you'll find one or more of them of interest. So first of all, New Zealand is in the southwest corner of the Asia-Pacific region, as you can see here from this map. It has an even smaller population and small GDP. So we're working under certain limitations financially, I suppose you might say, but we do have excellent scientists and clinicians here in New Zealand that are doing some wonderful work in this area. There are hidden depths to New Zealand. It's actually on top of, so geographically, there's hidden depths. It's on top of the 8th continent of the world called Zealandia. And we have a proud history of achievement in science through 3 Nobel Prize winners. And of course, the Australian John Eccles spent a considerable time in the University of Otago in Dunedin, where he pioneered the use of intracellular recordings in vivo. But that was then, and this is now. So in terms of New Zealand investment in neuroscience, we have, I suppose you might say, the standard kind of funding agencies like the Health Research Council and others. But in recent times, the government has also funded National Centers of Research Excellence, or COREs, and in particular, Brain Research New Zealand, and also the MedTech Core, which I will touch briefly on as well. So Brain Research New Zealand is co-directed from both Auckland and Otago. It's essentially the equivalent of New Zealand's Brain Research Initiative. It's been running for quite a few years now. Brings together lead researchers from a wide range across the country, as well as clinicians. We're tasked with undertaking research, but also training and community engagement. And this center is mostly around aging-related neurological disorders. So here's the network of centers, primary centers in this BRNZ network, including in University of Auckland and AUT, also in Auckland, University of Otago in Dunedin, and University of Canterbury and the New Zealand Brain Research Institute in Christchurch. So these local centers form the pillars, and of course, each of these centers have their own strengths. And I think particularly to focus or to note from an international perspective are in Auckland, the Human Brain Bank and Human Brain Cell Biobank, which are engaged in many international collaborative research programs. There's a very significant Parkinson's disease longitudinal research program in Christchurch, and also the Dunedin longitudinal study, which has been going on for 50 years now, from a cohort of 1,000 people in Dunedin who are now age 50 and obviously moving into the age of age-related neurological decline. So it's been a very significant study for both development and now aging. We also have established a national network of dementia research clinics across Auckland, Christchurch, and Dunedin, recruiting individuals with mild cognitive impairment or early AD and normal controls to do longitudinal work, collecting neuropsychological imaging and blood data, but also serving as a pool for testing interventions. And that's something that's still developing, but is using internationally recognized protocols so that data can be combined with other studies overseas. So we have lots of links internationally, including in this Asia Pacific region, but of course, obviously, plenty scope for more of these in particular. So the Brain Research New Zealand, as I mentioned, is aging-related focus, including everything from disease prevalence and mechanism biomarkers to more interventional and translational targets, which I'll touch on briefly now in the next few minutes, such as drug development, drug delivery, technologies, and actually communication, which I think is also important from a translational perspective. So just to touch on a few projects, I'll begin with one that I'm involved in, which is looking at gene therapy for Alzheimer's disease. We're particularly interested in the soluble amyloid precursor protein alpha, which we know from our work and many other studies is neurotrophic, neuroprotective, neurogenic, and enhances memory. We've shown that injections of this into the brain can actually enhance memory performance in aging and normal animals, but we need to find a translational way to get this in a disease treatment perspective. So we've been working on viral approaches to enhancing or reducing the phenotype in AD and models, mouse models. So we've been focusing initially on injections into the hippocampus. And we've had quite some success already with this approach using, first of all, a lentivirus approach with secreted APP therapy. We have shown already that this rescues water maze spatial learning in these APP PS1 mice that have been given this therapy, show enhancement of performance back to control level. And also the therapy rescues the animals from the impairment in long-term potentiation that is also seen in these animals. And then in another study using AAV approach, also into the hippocampus, we looked at neurogenesis, and as one of the results you can see yet unpublished, that the impairment and proliferation in the dentate gyrus in the AD mice is mostly rescued by the sapotherapy. So that's still not very translational. You're not going to inject virus into the brain. And so we also need to now are working on ways to try to get the virus in peripherally in a way and means such that it can get into the brain sufficiently to also rescue the phenotypes. And that is work that's undergoing currently. Another translational step, I suppose, is moving from rodent work to large animal models as well. And we don't have primate facilities in New Zealand, but we certainly do have lots of sheep. And my colleague, Stephanie Hughes, who I was doing the Alzheimer's work with, is also very interested in Batten's disease, which is a family of lysosomal storage disorders. And this is a developmental disorder, and the children very early on start developing behavioral changes, cognitive loss, visual impairments, eventually seizures, and premature death. And it turns out that a strain of sheep has been found that have a mutation in CLN5, a lysosomal protein, that has a very similar time course, or not time course, but overall phenotype over 2 years that looks very similar to Batten's disease, and is now serving as a useful model for studying treatment options. And again, Stephanie has been involved in using viral approach injected into the brain, into the occipital, parietal cortex, and lateral ventricle, into the lambs at either 2 or 3 months or 7 months, and to see if they can either rescue the phenotype or at least prolong the quality of life of these animals. And they've had some quite good success in this regard. So here is staining for CLN5 in hippocampus and cerebral cortex in normal wild type lambs or sheep. And then in the untreated ones at 23 months of age, very close to the time when they're actually going to die, you can see there's a lot of actual cell death and loss of staining actually for CLN5. But in the AAV9 treated sheep, you can see a rescue of quite a bit of the cells, prevention of the cell loss, and the expression of CLN5. And if you look at the brain size, you can see there's a good rescue there of the brain sizes and weights. But importantly, from a behavioral perspective, you can see that there's also a rescue here as well. And so over this time course of about 20 months, the ovine Batten's disease rating scale scores, you can see drop quite dramatically in the untreated sheep. But in the various kinds of viral treatments, including lentivirus and AAV9 virus injections, you get substantial slowing, at least, of the disease time course, although it doesn't cure it completely. And if you look at this behavioral kind of analysis of working through a maze to reach conspecifics, control lambs at 8 months of age can have very tight, clear evidence of going and mixing with their conspecifics, whereas in the untreated CLN5 negative mice, you can see that they have a much more disjointed pattern of behavior to get to the goal area. And at 19 months of age, then they have very little ability to work there through the maze, probably through a combination of cognitive and visual deficits. But again, with the AAV9 treatment at the same age, 19 months, the animals can work their way just about like with control animals. So there seems to be quite a bit of promise in this approach for gene therapy with this, at least in this model. And now they have been approved with an approach enabling clinical trials that are expected either end of this year or early next year. So as I mentioned, targeting therapies to the brain is quite an issue, and particularly for viruses, that's an active area of research internationally, I know, and we're involved in that. But there's other issues around therapies, getting the right therapies at the right time for the brain as well. So one of my colleagues here at Otago is exploring novel treatments for Parkinson's disease. So I suppose the problem with the current treatments, which is say levodopa delivery, is that you're maintaining a fairly level amount of dopamine in the brain, when in fact dopamine neurons actually tend to fire and burst during learning episodes and so forth. And dopamine replacement at the wrong timing seems to reinforce inappropriate movements. So how can we get a better approach to delivering dopamine drugs? So his approach that he's working actively on is packaging dopamine agonists into liposomes that actually also have a hollow gold nanoparticle attached to them, and then using ultrasound to be able to release the dopamine upon command. So they are now being able to achieve phasic release using pulses of ultrasound in sheep models of Parkinson's disease, 6-OHDA lesions, I believe, and they're getting some very good treatment effects with this approach. And of course, they're aiming to use this approach in humans as they work a bit further on developing the practicalities of getting that organized into human trials. So that's a novel approach for Parkinson's disease. And if I can move towards stroke as well, which is another aging-related disorder and can also lead to neurodegeneration over time in some cases. We know that, of course, like everywhere else in the world, stroke is a major neurological problem, particularly for the aged. And John is also interested in novel treatments here for helping with stroke recovery. So in this approach, he is, in rat models, he's been targeting providing stimulation to the contralesional hemisphere in the motor cortex and using low intensity intermittent theta burst stimulation to enhance stroke recovery in rat models with quite some success. So they're now working on in human studies to pilot the effect of contralesional burst stimulation to augment stroke therapy. So they have these arrays of electrodes that can be implanted neurosurgically subdurally and used to help and have shown that in some cases to enhance the effects of stroke therapy as well and promote recovery. So some quite novel approaches here in terms of the way therapies are normally presented. And finally, in this category, I'll just talk about the cochlea and hearing loss, because it is a major type of neurological disorder around the world. It's mostly due to sensory neural destruction in the cochlea, and there's very few approved therapies or good diagnostics or biomarkers to treat the damage that's going on in the cochlea from aging or for other reasons. And Professor Peter Thorne and the wider group of hearing specialists in the University of Auckland are working actively to get past this intratympanic route of drug delivery, which is the normal way of providing drugs into the cochlea through the round window. So without me being able to tell you the details at this stage, they're developing novel therapies to getting drugs applied for sustained release, to increase the round window permeability, and to distribute drugs throughout the cochlea to reach target tissues. And that's in collaboration with Brain Research New Zealand, but also the other core, MedTech core, which I want to just briefly go in and talk about now. So the other core, Center of Research Excellence in New Zealand, is devoted to technologies in a wide range of applications, including diagnostics and therapeutics, interventions, telehealth and informatics, assistive and rehabilitation technologies, tissue engineering, and so forth. So not all obviously are relevant to neuroscience, but some are. So here's some of the examples of some of the specific spin-off companies and approaches that have been developed through this particular core for breathing better, pelvic floor health for women, gut health, personalized orthopedics, and so forth. But more in our areas, you might look at VR technology for rehabilitation post-TBI, seeing improving screening for retinopathies, and rehabilitation for swallowing and eating impairments, which of course can also arise from neurodegeneration. And here's an example of a spin-off company that has been working on rehabilitation through virtual world simulations to help people learn to use their limbs to control movement through space in a virtual world and providing better patient outcomes in the real world. Another exciting approach from my point of view is using brain-computer interface to supercharge neuro rehabilitation, in this case again for stroke. And in this approach, as you can see, there's electrical stimulation applied to the leg at the tibialis anterior muscle at the time that voluntary or imagined movement occurs. When that is done by the individual that produces a signal in the EEG, which is called the movement-related cortical potential. And the idea is to time the exact stimulation to arrive at the brain at the same time that this movement is about to begin. And they have shown in this group, led by Professor Denise Taylor at AUT, that paired associated training with 50 reps of the voluntary or imagined movement plus the low intensity electrical stimulation of the muscle leads to substantial ability to increase the excitability of the motor cortex, and has serves as potential priming intervention to help with stroke rehabilitation, including improvements in muscle strength and voluntary activation in people with stroke. Then, of course, they have been moving to miniaturizing the EEG hardware and making it wearable, developing the software so that the MRCPs can be detected and used to help with the timing of electrical stimulation to the limbs as appropriate. So this has all been going underway for quite some time now, with one of the real advantages is that it personalizes the rehabilitation for the people suffering stroke. And of course, it's very acceptable to the patients and can fit with rehabilitation practice. And finally, my last set of vignettes is a slightly different area, which you might not normally consider as translational neuroscience. But we're very interested in, we believe that it's very important for information to be given by neuroscientists out to the community to help them understand what's going on in the brain and what they can do to reduce risk factors and promote perhaps recovery from injury or damage. And stroke is a very good example because most of the risk factors for stroke are all modifiable. And we understand through surveys that the understanding of the modifiable risk factors for stroke are very poorly understood across different ethnic populations in New Zealand, at least. And so Professor Valerie Fagan in AUT has developed an app, what's called the stroke riskometer app, where people input their relevant information and they can assess or predict the individual's risk of stroke in 5 to 10 years and recommend preventive strategies. And this very successful app actually has been endorsed by many of the major stroke and neurology associations around the world. So this is an example of useful technology to provide information to individuals on an individual basis. Another element of providing information in a translational kind of way to the community is by co-designing information platforms. In this case, it's developed by Nicola Kays, an MCI website resource. And through the co-design by interviews, developing workshops of what people need and want, and developing prototype websites and user testing, website refinement, and so forth, they developed a very interesting website which provides information to the individuals as that they hope will be useful. And I'm just going to just turn this. I'm going to play a brief snippet of this so you get an idea of what people say, the kind of information and how they want it delivered that people say they want. I'll just move along to...
Mild cognitive impairment, or MCI, is a relatively new diagnosis. Mild cognitive impairment, or MCI, is a relatively new diagnosis that clinicians use to describe cognitive changes, such as changes to your memory and thinking, that are greater than they would normally expect to see in a person of their age. Some people find it scary when they start to notice these changes, or when they are told they have MCI. However, most people with MCI are able to function from day to day and enjoy all the things they usually would enjoy by using strategies to manage those changes, such as a diary, keeping lists, and getting support from their family and friends. About 2 in 10 people will eventually return to their previous cognitive ability. Some people, about 1 in 10, may get worse and develop dementia. However, the majority will just stay the same. This website is a place for anyone with concerns about changes to their memory and thinking, not just people with MCI. This is where you can learn about how to keep your brain healthy, share tips and tricks for managing changes from day to day, or share stories about your experiences so that others can learn from them.
So that's just an example of a way of getting information to the general public through a website. And finally, I want to make one other point about getting information to people, and that is that the information that you have and the way you think about it and the way you want to present it may not be the best way to present it to people from different ethnicities. And so it's important to get information out to people in ways that they will connect with. In this case, of course, Brain Research New Zealand is very interested in getting information out to Maori, who are the indigenous peoples of New Zealand. This particular development has been involved in the development of an app for dementia for these reasons, that again, co-designing focus groups, the researchers have found that Maori want to find Maori ways of putting and naming and talking about Alzheimer's dementia, because it scares them, they don't know what to think about it. And if they had a Maori way of looking at it, then they would be much more receptive to the information that then follows about how to reduce risk. And so the term in Maori is mate wariwari, and that has been accepted by the Maori community. And therefore there is the co-design of this app using Maori friendly language and actual Maori language, Maori friendly graphics and sounds, videos of Maori speaking about mate wariwari. And again, involving co-design through consultation with whānau, with families living with dementia, and providing feedback. And this was just very recently launched and is being disseminated now out through New Zealand. Again, supported by the Brain Research New Zealand and the MedTech cores, where we're using technologies and information from both cores to inform the public. So again, as I say, not the usual kind of translational work that you might be thinking about, but also something that we think is very important component of the work that we all do. So in summary, what New Zealand and BRNZ bring to the international translational neuroscience effort is expertise in longitudinal studies and early biomarkers, molecular studies, imaging studies, clinical and so forth, evidence-based interventions, for which I tried to give you a few examples of the work that's being done in various domains. But we also have a collective of scientific excellence that's entwined with the community to bring understanding and education to those with neurological conditions and their families. And also shaping these efforts appropriately to match the cultural makeup of New Zealand society so that care and interventions can be accessible to everyone and address health inequalities, and maybe a model that other countries and organizations may wish to follow. And with that, I will close my talk and thank you for your attention, and be happy to answer any questions.
Thank you, Prof. Abraham, for such a great insight on aging-related research, such as dementia, Parkinson's disease, and stroke in New Zealand. And it's also great to also share some of the snapshots into your research. So that's great, a wide spectrum of approaches from animal research all the way to the usage of digital technology. So I'm just going to quickly go through the question Q&A box and just select. Perhaps I'll start off with Prof. Abraham. Could you comment on the challenges translating the research in small animals, such as rodent, to larger animals, such as sheep, and all the way into human? And how could we overcome those challenges?
Yeah, that's a very good question, and one that my own research program is, working with. And I suppose in the Alzheimer's field, where I've been doing most of my work, you know, injections into the brain are not translatable. And I think in terms of humans, for a start, there's just too many people, and it's an invasive technology, of course. And so this is why and also the brains, of course, in sheep and also particularly humans are much bigger than in rodents. So even what you might be able to achieve is probably not at all achievable with people people or sheep with larger brains. So this is why we're trying to find methods of doing peripheral administrations that can still target the brain. And I know that there's many approaches to this, breaking through the blood-brain barrier. But I think this is the wave of the future. And I know that people are working on this around the world. But I think targeting large brains, it will involve not directly treatments directly to the brain, but by peripheral administrations of the drugs or the viruses or whatever, and finding ways to get these treatments through the blood-brain barrier where necessary.
Great, thanks, Prof. Abraham. Just a quick question from Ankush Chakraborty. So obviously this is a question related to drug targeting in the brain. Could you briefly comment on the drug formulation such as Go nanoparticle being used? The nanoparticles? Yeah, no, I unfortunately, some of these things, there's some IP around some of these projects that I'm not allowed to disclose. So for that part, if you're thinking about the Parkinson's disease story that I was giving you with the nanoparticles, I can't comment, unfortunately. Actually, to be honest, I don't know myself. So so I can't help you on that question. But those who wish to find out more could write to Professor Reynolds and see what he might be able to share, if anything. Thanks, Prof. Abraham. A question from Dr. Shampa Ghosh. Is there any apps, so obviously this is about the technology, apps that help in early diagnosis in neurodegeneration due to obesity? This is something that we're interested in. I'm not aware of any myself, but other people may know, and that might come up in the chats, I suppose. There are aims to develop the app for Mallory to make it more like the stroke riskometer, where people can get individualized information about their risk for AD based on various modifiable risk factors. Of course, there's a certain genetic component which they can't do anything about. So that's on the cards, you might say, here in New Zealand, but it's some time away. And it could be obviously rolled out for anyone, not just Maori. And so there are plans to do that. There may be other apps around, but I'm not aware of that, of any offhand. Thanks, Prof. Abraham. So for the interest of time, I would like to thank Prof for your time, and once again, great talk and great insight, and yeah, a lot of great research happening in New Zealand, so that's very exciting. So I will now hand the floor over to Bexy to introduce the next speaker, and yeah, feel free to use the question and answer function chat to continue the engagement and question and answer. Okay, Bexy, off to you.
Thank you, Lin Kui. Couldn't agree more with you that Prof. Cliff Abraham successfully brought us tonight an eye-opening session about all the advancement happening in New Zealand. So from New Zealand, I would like to bring you to Malaysia. So I am happy to introduce you to the second speaker of the day. That's Professor Jafri Malin Abdullah. He is currently the Professor of Neurosciences and the Chairman of Brain and Behavior Cluster, School of Medical Sciences, University Science, Malaysia. He was awarded the prestigious Young National Malaysian Scientist Award in 1999 and awarded with the Top Research Scientist Award Academy of Science Malaysia in 2013. Part of his cluster's research are focused on new treatments in the field of neuro-oncology, medicinal chemistry in the field of CNS tuberculosis, epilepsy, and pain, as well as the neuroenopharmacology. Besides being responsible in setting up labs for functional magnetic resonance imaging, high-density electroencephalography, neurogenetics, even rodents, animal behavioral, electrophysiology, stem cell culture, and primary cell laboratories, his most recently research activities is in the field of neuroinformatics and neurotechnologies. He has also recently initiated Asia's first integrated Doctor of Neurosciences program, as well as the first joint psychology and cognitive neurosciences program. With that background, we know more about Professor Jafri Malin, and I would like to share his slide that he will then narrate through the presentation. And Prof, you have 25 minutes to share your insights with the audience today.
Thank you very much. Are you going to switch on the video to show my face?
I'm not sure whether I can do that. I think only you can do that.
Okay, I said yes. Okay, I'm on the video now.
All right, sure. So
I need
to share my screen.
Yeah, to see the PowerPoint. So hello everyone. My name is Jafri Malin Abdullah, and with my PhD student Muhammad Rida, we have prepared a presentation. And I would like to thank the organizers for thinking of us, inviting us to present today. The topic is Neuroscience Research 2015 to 2020 in Asia, Pacific, and Malaysia. How many have been translated to clinical use? Next slide. As we know, Asia Pacific is a huge area, and it consists of developed countries and developing countries. Next slide. And you can see that Asia Pacific countries they comprise of countries in East Asia, Southeast Asia, South Asia, and Oceania. And I would like to thank for the next few slides, Benny Ang from Scopus Singapore for this for providing us data to enable me to present this topic. So 45 countries contributed to index publication in Scopus, and as of 2013, Asia Pacific region, we spend about 2% of GDP on research, which is not very high, but we contribute 38.5% of world researchers. And the Asia Pacific contributed 4.5% of world research and development expenditures in 2017. Next slide, please. The population of Malaysia, I have to introduce Malaysia as well. Small country, population of 32.7 million. This is the estimate from July 2020. Our literacy rate is 93.7%. Our R&D is 1.44% of GDP. That was in 2016. And I think since COVID, it has dropped. The universities spend about 4.2 billion USD in R&D. We have 2,397 researchers per million people in 2016, where 51% of them are male and 49% of them are females. Next slide, please. Next slide. So I would like to discuss about the neuroscience publication output over the last 6 years, where with a little data from the early 2021, because most of the data we got was up to 2020 from the Asia Pacific region as defined by Scopus. And I would like to acknowledge again Mr. Benny Ang of Scopus Singapore for providing us this data. And as you can see from this histogram, you would see that China, of course, leads amongst all the Asia Pacific countries with 61,211 papers, followed by China, Australia, South Korea, India, Taiwan, Singapore, Hong Kong, New Zealand, and Thailand and Malaysia is placed at number 11. And you would see that China approximately contributes about 41%, Japan 15%, Australia 15%, South Korea 8%, India 7%. And the rest of the countries will provide about 2% of the neuroscience publication output in the last 6 years. Next slide, please. So you will see that this is a color coding showing the champions of neuroscience publication output over the last 6 years. And you would see that big countries like China, Japan, Australia, and Australasia, as well as India, leads amongst all the countries in Asia Pacific. Next slide, please. As we see the neuroscience population output, I would like to include, I included Malaysia as well as top 11, because we are the 11th country in the row. We would see that China has got at least 2-point-something times the output of Japan and Australia. And I think this is because the number of neuroscientists or scientists working in the field of neuroscience that publishes this paper. So we know very well in Asia Pacific that there are many countries, even like Malaysia, who publish thousands of papers, even in the field of neuroscience, but we are not sure whether they are cited. Next slide, please. So we look at the citation from these countries as well. Next slide, please. And we found that China has been steady, even with the large number of publications from China. We find that the citation is also great, followed by Australia, Japan, South Korea, India, and Singapore. And you can see that a country like Singapore, even though it does not publish as many papers as other countries, but the citation rate of publications from Singapore is very high and much, much better than countries like Malaysia, Thailand, and even New Zealand. So this is very interesting to see where our citation rates are going. We publish a lot, but our findings useful for translational neuroscience, or are they useful for something else? Next slide, please. So again, we see China as the big country that has lots of papers and has lots of citation, followed by Japan and Australia and India. Even Singapore has also a better trend than Malaysia. Next slide, please. So again, when we summarize the number of citations over the 6 years, we wonder what kind of quality of publications are coming out from other countries. As you might note that most of the citations from publications from big countries like China, Australia, and Japan, South Korea, and India, even Singapore, and other countries go to big Q1, Q2 journals, whereas countries down the road like Malaysia, even Thailand, and other countries may publish in Q3 or Q4 journals. Next slide, please. So as you see the citation per publication, we found it quite interesting because this was provided, next slide please, provided by Scopus Singapore. And lo and behold, we found a country like Tonga, small little island, has been cited more compared to other countries. And this is the value of when a country like, a smaller country like Tonga, Samoa, Papua New Guinea works with other developed country labs. So when they publish a paper, for example, in Lancet, and Scopus picks up all the countries, they do not note only the corresponding author or the PI of that project. They include all the authors from all different countries. So when a country like Tonga, this paper was from the Ministry of Health of Tonga, and you would see that this paper was highly cited. Next slide, please. So as we can see in this slide, we see that small countries like Tonga, Samoa and Solomon Islands, Papua New Guinea, a single paper would be able to be cited many times more compared to other countries, smaller countries. So this is very interesting to see that if a country or an underdeveloped country who wants to be put on the face of academia, they may have to work with other labs. They may have to work with other countries. A paper, and the paper is just not a data from Tonga itself or data from Samoa or Solomon Islands, but in that database is a collection of data from different countries in the world. So I think it will be much better in the future for countries in Asia Pacific to join forces. That's the message. If we do this separately, I think we will not be able to match the quality of publications from other countries. But if we join forces and combine data, especially in maybe clinical work or paraclinical work, where we work together in many centers, we will be able to come up with better data which can go to better journals. Next slide, please. So the authors per country, as you can see, next slide, shows that the China is again leading with 178,798 authors, and followed by Japan. Japan with 200 million people have about 446,700 authors in the Bay, classified by Scopus, writing up neuroscience papers. And you can see Malaysia as number 10, which has 2,980 authors which write papers in the fields of neuroscience. And you will see the other countries behind Malaysia, which have a number of authors ranging from 2,800 right to Marshall Island, where they have only one author classified as writing in the field of neuroscience. Next slide, please. So again, in this slide, it shows that the top 10 countries with a number of authors who write papers in neuroscience is again China, followed Japan, Australia, South Korea, India. So this is something that maybe the other developing countries in Asia Pacific can co-work or collaborate with authors from China, Japan, Australia, South Korea, and India, so that they are able to improve their quality of publications in the field of neuroscience. Next slide, please. So again, you see the 100 most cited authors comes from Australia, followed by China, Japan, Singapore, and the rest of the countries that are mentioned in this slide. So it is very interesting to see that the China has 33%. Remember that China had a lot of publications and they were right in front of the graph. And you find that Australia has overtaken them in the top 100 most cited authors. So I would like to just highlight a few authors that we found and highlighted to Scopus. For example, Professor Glenda Halliday. Next slide, please. From the University of Sydney, Australia. She has contributed lots of topics in frontotemporal dementia, and he has been highly cited by at least 142 publications. And she has now brought that bench to bedside via a recent paper and her project called Genetic Characterization of a Cohort with Familiar Parkinsonism and Cognitive Behavior Syndrome, a next generation sequencing study. So right now we see that sequencing study is coming in very importantly in the treatment of patients in the whole of the world. Next slide, please. In China, we see Professor Dingyang Shen. Professor Dingyang Shen was not only the top cited author, because other authors above him were mainly engineers, electrical engineers, talking about writing up on AI and neuroinformatics. But when we look at bench to bedside publications, we find that Professor Dingyang Shen contributed topics in the field of Alzheimer's disease and using computer-aided diagnosis. And his recent translational research was to bring deep neural network to differentiate brain activity between patients with first episode schizophrenia in healthy individuals, a multi-channel near-infrared spectroscopy study. So we see now the neurotechnology used to treat early schizophrenia. And this is a very interesting thing that we see coming to the 21st century. Next slide, please. From Japan, Professor Kenji Hashimoto from Chiba University, Japan, looking at a clinical experiment, ketamine, treatment-resistant depressive disorder and antidepressant. And now what he has brought to translational research was to look at trait dissociation as a predictor of induced dissociation by ketamine or esketamine in treating treatment-resistant depression. So, and he has done a randomized controlled trial in Japan. So this is quite interesting to see a ketamine, a very old drug, now brought forward to treat patients with depression. Next slide. And from Thailand, Professor Michael H.J. Mars, who looked into depression as well. And this was an animal experiment. Now he has brought it to randomized double-blind placebo-controlled clinical trial looking at a drug called pentoxifylline, a combination therapy in major depression disorder. So this is interesting when you can bring drugs from the lab into to the patients, to the hospital, to the bedside. Next slide, please. So translational research from bench to bedside is very important. Bedside has two meanings. It can be the bedside where the patient is in the hospital, or it can be a bedside in wellness where you're staying at home. So in our search in Scopus database with the keywords neuroscience, bench to bedside or translational, and restricted to Asia Pacific region from 2015 to 2021, yielded 156 papers. Initial abstract screening yielded 31 papers, and the animal clinical trials using mice, swine, primates, and zebrafish. Zebrafish is getting very popular in Asia Pacific region now as a source of bench experiment, as translational platform to bridge the gap between animal and human. Next slide, please. Among the advancement from bench to bedside in neuroscience is the use of non-invasive brain stimulation in Japan, in China, next slide, please. The drug therapy, psychotherapy, and neuromodulation in major depression disorders in China, South Korea, India, and Japan. And next slide, please, is another group in China, which are looking at implantable multi-electrode arrays, which was mainly used for electrophysiology in the lab. So now implantable multi-electrode arrays are coming into the human brain. Next slide, please. And research currently undergoing in Japan is a new technology in EEG. So this shows a picture of a non-human primate being experimented by the group in Japan. And very soon, I think we will have a very portable stick-on electrodes on our scalp, and this will be able to do many things for wellness and illness. And if you look at the brain interface, next slide, please, machine interfaces from China, and China is going great. You will see a lot of the publications from China now are focused on brain-machine interface. And China has got more brain-machine interface publications than countries like Japan or even Australia. So next slide, please. So neuroscience in wellness, we will see a country like Singapore, who has invented Neuro8. And Neuro8 is a capsule containing natural substances for many active ingredients, including 9 herbal extracts. They are not classified as medicine, but they're put under the Food Act. So this is now being sold all over Southeast Asia, and this has been supported by preclinical studies and clinical trials. Well done, Singapore. Next slide, please. And we see China, lots of Chinese papers looking at stem cell and brain injuries, looking at transfected mesenchymal stem cell, reduce neuroinflammatory response and maintain the integrity of blood-brain barrier, and also looking at learning and memory and reducing neuroapoptosis and improving neurological function through hippocampal neurogenesis and angiogenesis. Next slide, please. So good old Malaysia, you know, when I was looking for data to support Malaysia, I found interesting things. Number one, in the field of neurodegeneration and dementia, people have been doing experiments on Nigella sativa and the brain, but these have been published mainly in animal types of publications, but there's not been a clinical trial. But yet, N sativa is being used as a health mental brain supplement. Evidence from neuroscientific research on brain function boosts the intake of this herb. And this experiment was done by people from Saudi Arabia. So you see, the animal experiment was done in Malaysia, but the Arabs decided to use this more and support it by selling this off as food supplements. Next slide, please. Another interesting food supplement experiment for neurodegeneration is honey. So there have been different experiments done in Malaysia, done in especially animals, where it shows that honey improves spatial memory. And there is a compound called phenylalanine in the stingless beehive may trigger the upregulation of BDNF. And BDNF, as you know, is involved in synaptic function, and the upregulation of BDNF improves spatial memory in mice. The production and consumption of stingless bee honey has gained traction in Malaysia and has created a $1 billion industry without a single publication in a neuroscience journal in humans about human data. So we have yet to do a human study. Next slide, please. And also interesting, coming up in Malaysia is a company called Biotech, which has published a paper using brain-computer interface, using EEG for person identification. So EEG as a biometric system to identify a person has been started. This industry started in the island of Penang, and it has now moved to neurofeedback, gadgets for better sleep, transcranial direct current stimulation of the brain, all locally produced with support from China and also Korea, because the CEO, Mr. Peng Aikfong, was the person who's a psychologist who decided to do this outside of Malaysia and then bring the technology back to Malaysia and then improve translational neuroscience in Malaysia, which has been ongoing for the last 2 years. So thank you very much. Next slide. For listening to me. I'm open to question and answers. This is the data that was given to me by Benny Ng from Scopus, and I hope he has done justice. But it does show that we have lots and lots of things to do in Malaysia and in Asia Pacific is to catch up. But it's very difficult to catch up because with the richer countries like China, Japan, Australia, New Zealand, and even India. So the best is to work together. And if we work together, and we can see like countries like Tonga, Samoa, and Solomon Islands can achieve a good ranking by working together. So the message today's presentation is, let's work together. All the neuroscientists in Asia Pacific should work together and not be silos in their own lab. Thank you very much.
Thank you, Prof, for sharing a lot of data, especially the effort in collaboration with Scopus. And I do have one question from attendees here, particularly focused on this question. So is for the African scientific researchers, they always have interest in manuscripts in neurosciences, but they have major challenges to access the Scopus journals, which is costly. So, yes. Prof, from your opinion, is there any solution to this to help scientists access to those journals?
Yes, this is what Malaysia is trying to do, but we're still in discussion. We have so many universities in Malaysia. We also have so many universities in Africa, and trying to buy a license from Scopus for every single university is a lot of money. So if the Ministry of Higher Education or the Ministry of Health or the relevant university can have a giant prescription of Scopus journals, and then all the universities, let's say for example in this African country, they have 150 universities. Then these 150 universities can tap into the license of the Ministry of Higher Education of that African country and is able to get the full paper legally. No, we don't need to go to any Russian website or whatever to get these papers anymore. It's all done black and white. Malaysia is trying to do that now, but but because of COVID, I think this has slowed down a bit. Yeah.
Thank you, Prof. Another question is from what you have shared about the herbal extracts and its effect on the brain. So is there any universally accepted antidepressant that is from herbal extract?
Yeah, at the moment, there isn't any. So that's the problem, because you must remember that there are so many receptors on the surface of the neuron, and not even, don't count the neuron there, the astrocytes and all that. And one single compound will not work, not for psychiatric diseases. It has to be many compounds. And how do you model 25 compounds to cure a patient with schizophrenia? How can you allow a schizophrenia patient to swallow 25 tablets? Again, that is the question in neuroscience, in pharmacology. So how do we get a super compound that can dock into all receptors and give the same effect as if it has the effect of 25 compounds? Until now, we have not been able to solve this problem. That's why the drug industry for mental illness and also for maybe epilepsy, as we can see, has problems trying to solve and get the super duper compound that can dock on all receptors.
Thank you, Prof. So our last questions would be, is there any possibility, how do you see the possibility of establishing a brain bank in Malaysia? Like what Prof. Cliff Abraham have shared with us, this is happening in New Zealand and a couple of countries in Asia Pacific region. How do you see that happening in Malaysia in near future?
Yes. Let's not talk about first the brain bank. Let's talk about a simple biobank. There was a university in Malaysia who initiated a wonderful biobank, no brain tissue, just blood and other organs. And they had problems trying to maintain a very expensive entity. And there was just one institution with a certain number of samples. Just imagine if we need to have a bank for the whole of Malaysia, centrally placed in Kuala Lumpur, for example. And in one of those biobank, there is a brain bank, then it would be interesting to see, because we may be able to collect a brain tumor tissue. We may be able to collect blood from these brain patients, but to get organ donor, that means to say that the person who passed away donate his brain to science, it will be very, very tricky. I'm not trying to be sarcastic, but if you look at our tissue banking, you will see that a lot of the tissue banking come from Buddhists. There's always a joke, one Buddhist to save 5 Muslims. You know, so again, for the brain bank project, can do Muslims, do Hindus, do Buddhists, do other peoples of other races, do they want to donate their brain after they pass away for the sake of research? I am not sure. In Malaysia, I'm not sure. Singapore is doing it. I wish them well. I hope they succeed. But in Malaysia, it would be slightly tricky.
Thank you, Prof. That would be the last question for the interest of time. Thank you for your time joining us as the second speakers for today's webinar Horizon.
Thank you very much.
Okay, now
bye-bye.
I would like to pass the floor to you to introduce our third speaker. Thank you.
Thanks, Betsy, and thanks, Prof. Jafri, for your wonderful talk. Our next speaker is Associate Professor Amy Fu, and she's from the Division of Life Science at the Hong Kong University of Science and Technology. So she served several director and associate director roles, including at the Center for Stem Cell Research and the Molecular Neuroscience Center. Prof. Fu has published over 80 scientific papers. Her research interests are in understanding the signaling mediated by neurons and glia, specifically the innate immune pathway in synaptic plasticity and their dysregulation in neurodegenerative diseases. She recently identified genetic biomarkers of Alzheimer's disease in the Chinese population. And she also has extensive research experience on drug discovery and has collaborated with many pharmaceutical companies in undertaking those research. So today, Prof. Fu will give us insight for therapeutic development of Alzheimer's disease, and she will be focusing on the brain resident immune cells, the microglia. So Prof. Fu, the floor is yours.
Thank you, Lin Kuei. Thank you, Pixie, and also the meeting organizer for inviting me to share with you on our recent findings on the biology of Alzheimer's disease. So I would like to share the screen first. Okay. So the title of my topic of my presentation is Understanding Microglia's State Transition in Alzheimer's Disease, Insights for Therapeutic Development. So most of the funding I present today stem from the work done by me and my colleagues in Professor Nancy's lab at the Hong Kong University of Science and Technology in Hong Kong, China. So I thank Nancy for her support throughout the years. So Alzheimer's disease accounts for 60 to 80% of dementia cases. Older age is the most important known risk factor. Approximately 12.5% of people aged 65 and around 50% of people aged 85 suffer from AD. AD is increasingly large economic burden on the society. The current annual cost of dementia is estimated at $1 trillion, a figure projected to double by 2030. AD is an invisible pandemic, incurable and progressive brain disease characterized by memory loss, decline in reasoning and judgment, and impaired locomotion activity. So the pathology of AD is characterized by the presence of extracellular amyloid plaque deposition and the intracellular neurofibrillary tangles in the patient brains. Currently, there are around 5 FDA-approved drugs. All of them just provide the symptomatic relief. They all target the neurotransmitters. Hopefully, they can restore the neuronal communication among neurons. There have been no new drugs in the market for nearly 20 years. As of 2020, there were 120 agents that were in the clinical trials for AD treatment, while majority of them target amyloid or tau. Increasing in the number of agents target the other pathways, including neuroinflammatory pathway. Of course, the new therapeutic interventions are urgently needed. So this is the reason why we need to study the disease cause and identify the targets for the development of disease-modifying drugs. Emerging studies have suggested that innate immunity is important in the pathogenesis of AD. During AD development, there are various pathological changes occur in the brain, including the A-beta and tau pathology, reduced synaptic remodeling that result in the impaired neuronal communication, and finally result in neurodegeneration. In addition, emerging genetic and functional studies have suggested that microglia are key players in AD. As AD develops, the accumulation of A-beta in the brain activates the microglia, and the microglia attempt to eliminate the A-beta peptides through the phagocytosis and clearance. However, as more A-beta accumulate and the formation of the amyloid plaque, the microglia become dysfunction and disturb the innate immune homeostasis in the brain and cause the dysfunctioning of the brains. So it is important to investigate the molecular basis underlying the microglial dysfunctions in AD. There are recent studies of the single cell and single nucleus RNA-seq analysis of AD patients and transgenic mouse models, including our two recent findings, demonstrate that microglia are heterogeneous and the microglia change their cellular state upon disease progression. Here shows an example. In a transgenic, AD transgenic mouse model, homeostatic microglia can change to the subpopulation of disease-associated microglia as the A-beta plaque accumulate. So this microglial subpopulation exhibit a specific signature gene marker. They have a decrease in homeostatic gene signature, and they have an increase in the expression of them signature, including some of the AD-associated genes such as ApoE and CHAM2. So it has been suggested that the disease-associated microglia subpopulation with different activation state induced during the progression of the disease. So it is important to understand the roles of this microglial subpopulation in the disease pathogenesis. Nonetheless, the accumulation of A-beta as well as the chronic microglial dysfunctions result in the dysfunctioning in the brains, including the imbalance of inflammatory mediators, the dysfunctioning of the glial cells, the impairment of the synaptic plasticity and neuronal activity, as well as the neurodegeneration. All these dysfunctions lead to the cognitive impairment as well as the disease pathology. Therefore, we believe that targeting the cellular state transition of microglia may result in beneficial outcomes in AD. So our lab has focused to identify the molecules that can regulate the activity of microglia in Alzheimer's disease context. One of the protein that we focus is interleukin-33. It is a member of interleukin-1 receptor family. The protein binds to a heterodimeric receptor complex, including the ST2 and IL-1R accessory protein. Interleukin-33 acts as an alarmin. It is rapidly released upon cellular damage to maintain immune homeostasis. There are increasing links showing the association of IL-33 and its receptor ST2 signaling with Alzheimer's disease. Genetic variants of IL-33 and ST2 that are associated with AD have been identified. Moreover, the messenger RNA gene expression of IL-33 is found to be decreased in AD patient brains. As I mentioned before, IL-33 binds to ST2, the full-length receptor on the cells to trigger normal functions. ST2 indeed also can be expressed as a soluble form. This soluble ST2 is a decoy receptor to inhibit the normal function of IL-33 and ST2. We previously demonstrated that the soluble ST2 level in the blood increases in the individuals with mild cognitive impairment who have a high risk to develop into AD. Our latest studies demonstrate that soluble ST2 level in the blood and in the CSF of AD patients also increase. So the increase of soluble ST2 in the AD patients indeed suggests that there is a decreased activation of IL-33 ST2 signaling in AD. We also conduct experiments and demonstrate that IL-33 replenishment can exert beneficial effect in AD transgenic mouse. So injection of APPPS1 mice, one of the AD transgenic mice with IL-33 can alleviate the memory dysfunctions, improve the hippocampal synaptic plasticity impairment which is a major cellular mechanism that underlies the learning and memory. And also, IL-33 injection can reduce A-beta deposition. Here shows that the injection of IL-33 for 2 days can reduce the area of amyloid plaque deposition in the cortex of APPPS1 mice by 50%. So we then ask how IL-33 exerts its beneficial outcome in the transgenic mice. Given the impaired clearance of A-beta has been suggested to be one of the major causes of AD, so we examine whether the microglia responds to the IL-33 treatment. Here is an immunohistochemical analysis showing that there is an increase of IBA1 labeled microglia surrounding A-beta after IL-33 injection in APPPS1 mice. So this data suggests that IL-33 treatment can mobilize the microglia towards the A-beta. So we conduct a 2-photon in vivo imaging to examine the functional changes of microglia in IL-33 injected APPPS1 mice. Accordingly, we generate a transgenic line of APPPS1 that causes with CXVCR1 VYFP. So this APPPS1 expresses the fluorescent protein in the microglia. So here shows that the microglia are in green. And then we label the A-beta plaque with the fluorescent dye, which is shown in red. We then do the 2-photon in vivo imaging. So in the control condition, within the 12 hours, you can see that the microglia remain relatively stationary. However, after IL-33 treatment, you can see that this microglia can have a high mobility and migrate towards the amyloid plaque. So by doing the quantification, we shown that IL-33 injection can enhance the, can increase the microglial proportion with a high mobility. So here shows that for the proportion of microglia with high mobility changed from 2% to 11% after IL-33 treatment. So when you just look at the microglia with high mobility, we show that they are migrating towards the A-beta plaque. Moreover, we also demonstrate that in this microglial subpopulation that respond to IL-33, they have a higher A-beta phagocytic capacity. So we label the A-beta with methoxy X04, which is fluorescent dye to label the A-beta. And then we treat the, and then we inject the mice with IL-33. We isolate the microglia by the CD11B and CD45 antibody and subject to a cell sorting analysis. Here you can see that IL-33 treatment can increase the number, the percentage of microglia with A-beta uptake. So we ask the question, how does IL-33 stimulate this specific microglial subpopulation? And what are their molecular signatures? And how do they promote their A-beta clearance? So it has been suggested the transcriptome profiling can regulate the functions of the microglia. So we conduct a global RNA seq analysis in the microglia isolated from IL-33 injected APPPS1 mice at different time duration. Here, interestingly, you can see that the transcriptome profile of the microglia actually is regulated in a stepwise manner. So here you can see that at the 3 hour, there is a there is a group of genes that increase at the 3 hours IL-33 treatment, and when we look at the gene ontology analysis, we shown that this group of genes are associated with the stress response. And interestingly, for this panel of genes, they are the genes that associate with chemotaxis. These genes are increased at the 3 hour treatment, IL-33 treatment, and remain elevated at the 8 hours. To further look into how IL-33 regulate the functional response of IL-33 microglia subpopulation, we conduct a single-cell RNA-seq analysis in the IL-33 treated APPPS1 mice. So here you can see that we do the T-layer analysis, and we found that actually the disease-associated microglia are clustered in at a similar manner between the control and IL-33 condition. However, when we look into the gene signature in a subpopulation of DAM, we found that there is an increase in the expression of genes that are associated with the MHC2 genes. And there is a restoration of the gene signature, the homeostatic signature. MHC2, which called the major histocompatibility complex class 2 genes, it was known to be microglial activation marker. So we labeled this subpopulation of microglia as IL-33 responsive microglia, or in short, IL-33-RM, which express the cell marker MHC2. So we then want to understand what is the interaction or what is the functional response of this specific microglial subpopulation in APPPS1 mice. So we conduct an immunohistochemical analysis. Interestingly, we found that the IL-33 induced a specific subpopulation of microglia that express MHC2 genes. So they highly colocalized with the amyloid plaque. So the IL-33-RM is co-labeled with MHC2 and IBA1, which stay labeled in yellow, and the A-beta plaque is shown in blue here. We also compare the A-beta phagocytic ability of this MHC2 positive microglial subpopulation with the other non-MHC2 expressing microglia. Here, we also show that they possess a higher A-beta uptake capacity. As far, we demonstrate that IL-33 injection can promote the A-beta clearance, and at the same time, they can change the cellular transition in the microglia. So here we show that IL-33 injection can change the homeostatic microglia to the chemotactic microglia, which express a higher chemotactic ability towards the A-beta. When they interact with the A-beta, they also adopt a phagocytic state, which are characterized by an increase in the A-beta clearance ability. So we suggest that IL-33 exerts their beneficial outcome through the induction of IL-33-RM, this specific MHC2 expressing microglial subpopulation. So given this IL-33-RM has a distinct set of signature genes. So we asked how to regulate the gene signature of this microglial subpopulation. It was suggested that the transcriptome profile of microglia are regulated by the epigenetic remodeling. So we ask next question. So whether the epigenetic landscape of IL-33-RM is regulated. So we examined the genomic regions in this microglial subpopulation, whether which genomic regions has a higher chromatin accessibility. So we conduct chromatin accessibility analysis using the ATAC-seq. So we compare the chromatin accessible region signal in the IL-33-RM with those in the non-IL-33-RM. So the ATAC-seq analysis shown that in the IL-33-RM, there is an increase of chromatin accessible regions in specific set of genes. So when we conduct the gene ontology terms analysis, we shown that actually the genes that have the increased chromatin accessibility actually are associated with the genes that have the myeloid cell differentiation, the myeloid and the macrophage activation. We then further look into the chromatin accessibility at the regulatory regions of the MHC2 genes that we shown it is the cell marker of IL-33-RM. We shown that actually in IL-33-RM, the chromatin accessibility signal increases in this in the regulatory region of these MHC2 genes. Increased chromatin accessibility allows more transcription factor binding. So we ask another question. So which transcription factors bind to these regulatory regions? So we perform a motif analysis in these chromatin accessible regions and found that actually these chromatin accessible regions are enriched with myelination specific transcription factor, the PU1. So it suggests that PU1 can bind to these chromatin accessible regions of the microglia genes. So we then conduct chipset analysis using the PU1 antibody. So we demonstrate that PU1 indeed binds to this regulatory region of the MHC2 genes in the microglia in APPPS1 mice after IL-33 injection. Of course, you will ask whether the PU1 activity is important for the IL-33 induced increase of IL-33-RM and the A-beta clearance in the transgenic mice. So we inject a small molecule inhibitor of PU1 that can block the interaction of PU1 and the DNA before the injection of IL-33. We then examine the induction of IL-33-RM in these mice and also examine the A-beta pathology in these mice. So here you can see that when we treat the mice with IL-33 alone, you can see that actually there is an induction of the DAM of the IL-33-RM signature. So you can see that there is an increase of MHC2 genes and the restoration of the homeostatic signature. However, when we co-treat the mice with the DB2313, you can see that there is an abolishment of the induction of IL-33-RM. Moreover, when we look at the A-beta deposition pathology in the transgenic mice, you can see that in the IL-33 treated condition, when compared with the velical control, there is a significant reduction of the A-beta deposition. However, co-treatment with DB2313, you can see that there is an abolishment of the beneficial effect of IL-33. So in general, we demonstrate that IL-33 induces a microglial subpopulation, IL-33-RM in APPPS1 mice. And the remodeling of the chromatin state and transcriptional activity of PU1 controls the induction of IL-33-RM through the increase of MHC2 genes and the restoration of homeostatic signature genes. We further shown that IL-33-RM is important for the A-beta clearance and reduction of amyloid pathology stimulated by IL-33. So we also try to examine whether IL-33 ST2 signaling is deregulated in AD patients. So we start to conduct some human analysis. So we perform the whole genome sequencing and the GWAS analysis in AD Chinese cohort with the plasma soluble ST2 data. So we've shown that the regulation of the plasma soluble ST2 indeed is contributed by the genetic components. So here we've shown that some genetic variants in the IL-1, IL-1 locus that encode soluble ST2 and ST2L are associated with the plasma soluble ST2. So here you can see that for the individuals that carry 2 copies of this soluble ST2 variant, they show a lower plasma soluble ST2 level. Interestingly, we had a collaboration with Professor Colin Masters. So we examined the effect of this variant in the AIBL cohort. And interestingly, we observed that this soluble ST2 genetic variant seems to exert a protective effect in the progression of AD pathology. In a subgroup of AD patients, you can see that the patients that carry 2 copies of this soluble ST2 genetic variant have a slower decrease in the brain volume compared to the non-carriers, as well as the carrier that only have 1 copy. So it will be very interesting to further examine how the soluble ST2 genetic variant regulates the pathology of AD patients. So together, we demonstrate that in AD, the microglial dysfunctions lead to the disturbance of the brain homeostasis and the brain dysfunctions. So we demonstrate that the activation of the IL-33 ST2 signaling or the modulation of the beneficial functions of microglia can decrease the A-beta deposition and inflammation, thereby enhances the synaptic communication and memory functions. So we suggest that the identification of multiple targets along this pathway may be important for the development of the therapeutic strategies for AD. So I would like to thank the group member in Professor Nancy's lab and also all our collaborators throughout the years. And we also like to thank the funding agencies for throughout the years. So this is the end of my presentation. Thank you very much.
Thank you, Prof. Femi Fu, for this educational introduction on Alzheimer's disease and also very detailed insight on how microglia could be targeted for Alzheimer's disease. So I've got a couple of questions already in the Q&A. I think I'll just sort of quickly start off with, so this is sort of a chicken or the egg first question. So what is driving the neurodegeneration in AD? So are they sort of, what comes first, the microglia dysfunction or amyloid beta accumulation, or it's sort of a good interplay between those two?
Okay, so given the familial AD diseases, actually, we know that there is an accumulation of A-beta. And I always go back to ask, so whether there are any genetic relations. So given for the late onset Alzheimer's or the majority of Alzheimer's disease, the genetics are associated with the dysfunctioning of microglia. I believe that it is associated with the dysfunctioning of microglia to cure the A-beta or the other toxic substance in AD. So microglia dysfunction may be a major mechanism that mediate the pathogenesis of the disease.
Thanks, Prof. Amy. Talking about clearance of amyloid accumulation in the brain, could you comment a bit on the recent found lymphatic system or the lymphatic system in the brain? So do they also play a major role on top of what microglia is doing?
I think it is interesting to look at the lymphatic system. So given there are more studies suggesting that there are interaction of different glial cells in the brain, including the astrocyte microglia and even the involvement of endothelial cells. So definitely, it is an interesting topic that we should look into. And I can also introduce, we recently published a paper in PNAS and suggesting the endothelial cell dysfunctions using is maybe associated with the AD pathology, given we shown that the molecular signature of the endothelial cells was deregulated in the AD patients.
Great, thanks, Prof. Amy Fu. Another questions from the participant. So I think we've got a few people asking this, Poonam and Miwako Ozaki. So you've talked or you've gave a lot of information or data on the IL-31, and I think most of them are in the preclinical level. Could you comment on how this IL-31 could be used as a therapy in human population or in AD patients?
Okay. So I think IL-33, we have a quite solid evidence at the preclinical level to show that they can mobilize the microglia, but when it need to be moved to the next level. Of course, we need to look at the benefit of IL-33 in a holistic view. So we are working on that. Indeed, we licensed the technology to and collaborate with a company to examine the therapeutic potential of this approach. But I think other than the working on the IL-33, as I mentioned, given in the body, there is a soluble ST2 protein that acts as endogenous inhibitor. So targeting the endogenous inhibitor also is an alternative to regulate the microglial functioning in the disease. So we have different approaches to regulate the microglia functions in the disease. That is the direction that we are working on.
Great. Yeah, so we're really looking forward onto this work translating from preclinical to human, so very exciting. So in addition to IL-33, so there are other inflammatory markers that's also involved in Alzheimer's disease pathology. So there's this question from Saron Kumar Das. So could you perhaps comment a little bit on the interferon response, the type 1, type 2 interferons response?
Okay, so I think in general, indeed, the actually in the past, there are some cytokines have already been suggested to regulate the AD pathology, or they have been studied to whether they can be the targets to be modulated in the Alzheimer's disease therapy. So for the cytokines, like IL-33, we demonstrate its effect interleukin-1, interleukin-12, 23, or interleukin-10. So in the past, the clinical trials used by modulating the cytokines are not quite successful. I think the reason is that because the understanding of the effect of this cytokine in the microglia is not as well studied as up to date. So actually, I think for the cytokine modulation, we definitely need to apply it at a very appropriate state, because you know, there is actually the cytokine can work together, may counterbalance the effect of the cytokine. So we need to do more research to understand the roles of cytokine in this microglia functioning in the disease.
All right, that's great. Thanks, thanks, Prof. Amy. So I think for the interest of time, I think great talk, and I think there might be one or two more questions. So Prof. Amy Fu, you could potentially just type a response to the answer on the Q&A chat. All right, I will now hand the floor to Pixie to introduce the next speaker. Thank you for your presentation, Prof. Amy.
Thank you, Amy. Thank you, Lin Kui. Hi, attendees. Thank you for staying with us. Now we would like to welcome Professor Michisuke Yuzaki from Japan. So let me check whether Prof. Yuzaki is can start your video. Okay, great. We see you. Thank you, Prof. Nice meeting you. So I would like to begin his talk by having a brief introduction about Prof. Yuzaki. He is currently the professor and chair of the Department of Neurophysiology, Kyoto University School of Medicine. Prof. Yuzaki's research goal is to understand molecular mechanisms that control synapse formation, elimination, and maintenance, focusing on new synaptic organizers, the complement family proteins, such as C1q and C1q-like proteins. He is also interested in how neuronal activities can regulate functional synaptic plasticities, such as LTP and LTD, at excitatory synapses. His lab also developing optogenetic tools that can directly and reversibly regulate LTP and LTD at specific synapses to clarify their roles in vivo. Through these studies, he and his group wish to contribute to a better understanding of mechanism underlying neurological and neurodevelopmental disorders, including the Alzheimer's diseases and autism. For that, I would like to pass the floor to Prof. Yuzaki to bring you to his research world, some exciting breakthrough from Japan. Thank you, Prof.
Can you hear me properly?
Yes.
Okay, good. Okay, good morning, afternoon, or evening, ladies and gentlemen. And I'd like to thank Pixie for kind introduction. It's really a great pleasure and an honor to give a talk at this second Global Neuroscience Horizon webinar series organized by IBRO and FOUNDS. Today, instead of introducing translational research trends in Japan, I would like to talk my own research, which I think will showcase how basic neuroscience could be translated into clinical medicine. Okay, using various imaging techniques, we can now visualize a gross connection between different brain regions in living human. And as you know, compared to normal people, many neuropsychiatric or neurological diseases such as schizophrenia, autism spectrum disorders, and Alzheimer's diseases, patients show a hyperconnectivity and sometimes hypoconnectivity between certain brain regions. So for this region, many of these diseases now referred to as connectivity disorders or synaptopathy because they have problems at the synapses. Then the important question is how such connectivity changes occur. I think it is basically mediated by functional connectivity changes, which are known as LTP or LTD, long-term potentiation, or long-term depression, the functional levels. But for the long-term changes, probably structural changes also occur in our brain that changes the wiring between several brain regions. So these changes are modulated by molecules called synaptic organizers. So today, I'd like to talk about two topics. First, I'd like to introduce you the new concept, extracellular scaffold proteins. This is a new family of synaptic organizers. And then at the second part of my talk, I'd like to explain how the findings can be translated into more clinical usage. So in the last maybe 15 years, many synaptic organizers have been identified. These are mainly classified into two categories. One is the cell adhesion molecules that directly connect presynaptic neurons with postsynaptic neurons. Basically, these are cell adhesion proteins such as N-cadherin or neurexin or neuroligand or LLRTM. I will talk about neurexin later, which is located at the presynaptic site. And the second category is secreted molecules, which are secreted from neurons as well as glia. And they diffuse and instruct neurons to make synapses around here. Members that belong to this family include Wnt, FGF, TGF-beta, or GDNF. And the third category, which I'd like to mostly talk about, is the what we call extracellular scaffold proteins. So these are also secreted from neurons or glia, but they do not diffuse but stay at the synaptic cleft as an extracellular matrix and provides scaffold that to accumulate presynaptic membrane proteins or postsynaptic membrane proteins. The typical members of this family is like Amy introduced, belong to innate immune system, C1q family proteins, and also neuronal pentraxins. And glia also secrete some ESP family proteins such as transpondin and heavin. So what is the C1q family? I think most of you have already heard about them, but the C1q family is characterized by the C-terminal globular C1q domain, which make the trimeric structure, and many members of this family make a higher order margimeric structures using various motifs. And prototype member of this family is, of course, the C1q itself, complement C1q, which belongs to the innate immune system. C1q recognize many foreign objects to remove them out of our body system. And interestingly, members of this C1q family, which at least we found in our genome 32 members, are highly expressed in the central nervous system, and some are highly expressed in the adipose tissues, and they are mostly secreted, and in the central nervous system, they regulate synapses. And one of the features of the C1q family, or ESP, extracellular scaffold protein, is that they are potent synaptogenic activities in adult brains in vivo. So I take CBN1 or cerebellin as an example, because this was the prototype of the C1q family in the central nervous system. As the name implies, CBN1 cerebellin is highly expressed in the cerebellum, mostly selectively in granule cells. And granule cells make synapses, excited synapses with Purkinje cells by its axon called parallel fibers. So this is a regular glutamatergic excited synapses. And interestingly, CBN1 is highly enriched at this synaptic cleft. So when we knock out CBN1, what happens is that the reduction, severe reduction of the number of parallel fiber synapses, up to 80% of synapses are reduced. And because of that, animals show ataxia. So this is the work we originally started in collaboration with Jim Morgan when I was still in the US. So CBN1 knockout mouse show ataxia. So when you put these animals on the rotating rod, they cannot stay on the rod. because of the loss of the synapses. But interestingly enough, when you inject CBN1 proteins, recombinant proteins directly into the cerebellum, just 2 or 3 days later, we saw the almost complete recovery of the synapses, and then also the same this same mouse can now stay on the rotating rod almost forever until she's really tired. So this is a movie that clearly shows the potent synaptogenic activities of CBN1 in vivo. So then how CBN1 are produced in the granule cell, parallel fibers can be localized at the synaptic cleft. So in other words, how is CBN1 released? So to address this question, we express CEP-tagged CBN1 together with M-orange2 tagged cathepsin B. CEP and M-orange2 is a fluorescent protein whose fluorescence signal is highly sensitive to pH. So when the pH surrounding the CEP or M-orange2 is low, the fluorescent signal is very weak. So this is the axon of cultured granule cells. And what we found was that when we increased the neuronal activities of granule cells, we saw the increase in CBN1 signals along granule cell axons, which is co-localized with cathepsin B and M-orange2 signals. As you know, cathepsin B is a lysosomal enzyme located in lysosome in axons. So because inside the lumen of the lysosome is acidic, the signal is low. And CBN1 seems to be localized in the lysosome in the axons. And exocytosis of CBN1 was selectively observed in axons, not in soma or dendrites of granule cells. And because cathepsin B is a lysosomal enzyme, it actually degrades extracellular matrix around the release site, which is probably necessary for the morphological changes that are induced by CBN1 release, and namely the presynaptic Bouton formation. So for the second question, what are receptors for CBN1? We make a long story very short, but we found that it is the delta 2 family of glutamate receptor expressed in cerebral Purkinje cells. And by binding to GluD2, CBN1 can accumulate various postsynaptic proteins. And at the presynaptic site, CBN1 also binds neurodegeneration, which is a cell adhesion type of synaptic organizers, and accumulate synaptic vesicles, and eventually induce presynaptic Bouton formation, as I explained in the previous slide. So CBN1 serves as the bidirectional synaptic organizers at parallel fiber Purkinje cell synapses in the cerebellum. So GluD2 was actually originally found by Michina's group and Seberg's group independently back in 1993. And because it is similar, its amino acid sequence is very similar to glutamate receptor. It's been referred to as glutamate receptors, but it doesn't bind glutamate. So it was called orphan receptors. So after 17 years, 70 years birthday, they orphanized the glutamate receptor. So CBN1 is the ligand that binds to glutamate receptor delta 2, GluD2, and serve as a synaptic organizer. So I told you about the CBN1's role in the formation and also the maintenance of granule cell Purkinje cell synapses in the cerebellum. But recently, Matt Anderson's group also showed that CBN1 is also expressed in certain brain regions in the forebrain, such as ventral tegmentary area. And interestingly, in chronic epilepsy mouse models, which show increased neural activities, CBN1 messenger RNA expression was shut off in the VTA, and because of the lack of CBN1 at VTA nucleus accumbens synapses, those animals show autism-like phenotype, ASD-like symptoms. So CBN1 can also work as a synaptic organizer in certain brain regions, in addition to the cerebellum. So here's a summary of the first part of my talk. CBN1 is a very strong synaptic organizer that belongs to the extracellular scaffold protein family of synaptic organizers. And CBN1 forms a tripartite complex and bridges presynaptic site and postsynaptic site by binding to neurodegeneration and GluD2. And one of the interesting features of the CBN1 as a synaptic organizer is that its activity dependency at the acute state increase in neural activity induces release from axonal lysosome. But after chronic neural activity, messenger RNA expression is suppressed and synapses are reduced. So let me move on to the second part of my talk. So how can we make good use of these findings into clinical medicine? So as I told you, CBN1 serves as a very strong synaptic organizer that fiber Parkinson's synapses in the cerebellum by binding to neurodegeneration and GluD2. But the problem is that GluD2, its innate postsynaptic receptor is highly enriched in the cerebellum. So we wanted to have a synaptic connector that can work outside the cerebellum. So, we focused on another extra synaptic scaffold protein, neural pentraction, which is known to accumulate postsynaptic AMPA receptors. And AMPA receptors is everywhere when excitatory synapses are formed. So NP1 can serve as a synaptic connector, but at least in vivo, it seems that the NP1 does not have a good presynaptic receptor. So we decided to use the neurodegeneration binding site of CBN1 here and AMPA receptor binding site of neural pentraction 1 and make a new synthetic chimeric synaptic connector called CPT. So we expected that CPTX can induce excitatory synapses by binding to AMPA receptors and neurodegeneration. And to achieve this goal, we first identified neurodegeneration binding site of CBN1 in collaboration with the structural biologist Radu Arstes in LMB. And then we recently determined the AMPA receptor binding site of neural pentraction 1 called PTX. So then we could combine CRR and PTX to form a designer synaptic connector CPTX. And first we injected CPTX into GluD2 knockout mice. And unlike CBN1, which cannot show any synaptogenic effect in the absence of GluD1, we expected that the CPTX can induce synapses by binding AMPA receptors in GluD2 knockout mice. So this is a typical gait of the GluD2 knockout mice. As you can see, these animals show highly ataxic gait. And 3 days after injection of CBN1, CPTX into the cerebellum of the GluD2 knockout mice, we saw the improvement of the ataxic gait, although they still show ataxia. But by measuring 3 parameters, namely stride length and stride irregularity and the gait speed, all 3 parameters showed a significant increase by CPTX injection, but not by CBN1 injection into the cerebellum of GLUD2 knockout mice. So it looks like CPTX is working in the absence of GLUD2. So how about hippocampus or other brain regions? So next we use the Alzheimer's disease model called 5XFAD mice. These mice carry 5 familiar Alzheimer's disease genes. And we examine the first structure, excited synapses in the CUA1 pyramidal neurons by Golgi staining. And spines are shown by red colors here, red dots here. And compared with wild-type animals, as reported previously, 5XFAD mouse models show reduced spines or reduced excited synapses that make synapses onto spines. But 3 days after injection of the CPTX into the hippocampus, we found increase in the number of dendritic spines in 5XFAD mice. So this is the quantified data. Compared with wild-type animals, the number of spines is reduced in 5XFAD mice that received saline. But when CPTX injected 5XFAD mouse showed almost normal dendritic spine numbers. And I skipped most electrophysiological data, but I just show you the miniature EPSCs taken from acute slices prepared from 5XFAD mice that received CPTX injection 3 days ago. As you can see, compared with mock injected animals, the frequency and also the amplitude of miniature EPSCs in CA1 pyramidal neurons were increased without changes in the miniature iPSC frequency or amplitudes. These results indicate that the effect of CPTX is specific to excited synapses. And we also found that the LTP impairment in 5XFAD mouse was restored, rescued by CPTX, and also evoked EPSP amplitude was also restored by CPTX. I just skipped the detail, but I like to go on to the behavioral data and also skipping the maze analysis, but I show you the fear conditioning. So in wild type mice, when you place in context A and apply electrical shock, next day they remember the context. So when animals are in the context A, they show more freezing than in different context B. This is normal fear conditioning, context fear conditioning. And however, in 5XFAD mouse, as reported earlier, they show generalized fear to both contexts. So they cannot discriminate context A from context B. And when we injected CPTX into the hippocampus of 5XFAD mouse, we found the almost complete recovery of the context fear memory, which is known to be dependent on dorsal part of the hippocampus. So how about other brain regions? So we next examined the effect of CPTX in spinal cord injury models. I just show you the hemisection model, but we found a similar effect in contusion model. So we applied hemisection at the level of thoracic 5, and because of that, those animals show complete paralysis in the hind limbs without any effect on the forelimbs. And 1 week after hemisection, we injected CPTX or CBN1 directly into the spinal cord. And y-axis shows a BMS score. 0 indicate complete paralysis. 9 is completely normal. So with a saline injection, those animals show some recovery after 8 weeks. CBN1 injection was not effective at all, unfortunately, because GluD2 is not highly, the receptor for CBN1, GluD2 is not highly expressed in the spinal cord. And as we expected, CPTX injection significantly facilitate the recovery from the complete paralysis. And that effect lasted more than 8 weeks. So this is so you can see two animals, and animals which has tails with black color is a control. So this is a control. And tail with red color is a CPTX injected animal. And please look at the hind limbs. These guys, control animals, show almost complete paralysis at 4 weeks after injection, but red ones can use hind limbs almost, very well. There's still some defect. So what was happening at the level of the spinal cord? So we examined immunohistochemically after injection of CPTX into the spinal cord, this region. And we stained presynaptic receptor neurexin with blue and postsynaptic receptor AMPA receptor gluea in green, CPTX with a pink color. As you can see, in many regions, we saw that CPTX signals are flanked by neurexin and gluea, indicating that CPTX really promoted the formation of synapses at the spared intraspinal networks. And we didn't see any evidence supporting the reconnection at the section, at the region, but the spared interspinal networks were observed here. And why the effect could last more than 7 to 8 weeks? So we found actually after 1 week, injected CPTX was completely degraded. It's no longer there. But what we found is increased synapses even at 7 to 8 weeks later. So these results indicate that CPTX helped, promoted the initial synapse formation and endogenous synaptic organizers may take over, may replace the original synapses formed by CPTX. So we developed a designer synaptic connector called CPTX, which consists of endogenous synaptic organizer CPAN1 and neuronal pentraxin 1. And CPTX could induce excitatory synapses, not only in vitro, but also in vivo. And more importantly, CPTX can treat 3 disease models. It could treat ataxic gait in GluD2 knockout mice, and also it could restore the impairment in context sphere memory in 5XFAD. And I didn't show data, but it also improved the spatial memory observed in 5XFAD mice. And it greatly promoted the recovery of the locomotion in spinal cord injury models. The good news is that the CBL1 has a member, 32 members. So this is a CBN1. And all these members are involved in synapse formation and maintenance of certain neuronal circuits. And this is a C1QL subfamily. And C1Q itself is involved in synapse elimination, especially during development and also during diseases. And we found that the C1Q, like 2 and 3, are involved in the recruitment of postsynaptic kainate receptors at the MOSI fiber CA3 synapses in the hippocampus. And C1Q, like 1, which found is involved in the formation of climbing fiber Purkinje cell synapses in the cerebellum. And Tom Shudoff's group also showed that C1QL3 is involved in the formation of synapses between basolateral amygdala and medial prefrontal cortex. And because all C1q proteins and most extracellular scaffold proteins are made of modular design, that means we can design more connectors once we know the structure of these connectors, molecules. So we hope we can understand and hopefully develop new treatment based on targeted at synapses based on the synaptic organizers. And that can be useful for the connectivity disorders or synaptopathies such as Alzheimer's disease or ASD or bipolar disorders. So with this, I'd like to lastly thank my collaborators. Masahiko Watanabe and Kenji Sakimura are long-term collaborators. And Dr. Takeuchi performed all experiments in spinal cord injury models, and hippocampal models experiments were done by Dr. Alexander Ditaev, and structure biologist Radu Arsex helped to design and produce CPTX. And this is my laboratory members. Thank you very much for your attention.
Thank you, Professor Michisuke Yuzaki. So thanks especially particularly on the last few slides, because there has been one questioning about how about the families members for cerebellum, because you'll be highlighting is cerebellum 1. Then there's question about how about 2 and 4. So you just answer it timely for the last few slides. Perfect. And here I have another 2 questions from the attendees. So I'm going to read it out now. So first is, how do you summarize the function of cerebellum 1 mediating to synapse by acute and chronic activities? Do you think cerebellum 1 doesn't contribute to stable connection at all?
Maybe I could make myself understood very well, but CBN1, for example, when you knock out in adult, you lose synapses. That means CBN1 is essential for the maintenance. It must be always there. So in other words, when you have chronic neuron activities, such as during epilepsy, you may lose synapses because you lose CBN1 messenger RNA transcription completely. It may be a way of safeguard to prevent excessive stimulation of excited synapses. I hope this can answer this question. And acute effect is mostly, I think, occur during development. So, and I'm not completely sure whether the secretion is always dependent on neuronal activity after circuits are mature. So all these experiments I showed you were done during development. So I don't know, the acute secretion really occurs in
Yeah, so I think I will advance you with the last questions for your presentation. Is there an interconnection between BDNF and Prop-BDNF on one hand with the cerebellum 1 in regards to LTP and LTD synaptic plasticity?
Oh, this is a bit difficult question. Yeah, BDNF, one of the major unanswered questions in this field is that there are so many synaptic organizers. We don't know how each synaptic organizer interact with each other and which comes first. And everyone who found particular synaptic organizers want to claim that they are more organized that comes first, but I'm not really sure. So there might be interaction between BDNF, TrkB, and CBN1 will lead to, for sure. And main difference is that the CBN1 is released from presynaptic terminal, not from the postsynaptic terminals, dendrites at all. But the BDNF is mostly secreted from postsynaptic side. Although there are some reports showing that the BDNF can also be secreted from presynaptic side, but I don't think that's a major pathway.
Right, thank you. There will be a few more questions, but I'll leave it to you to answer to the attendees after that. So Lin Kui, I would like to pass the digital floor back to you to introduce the fifth speaker of the day. Thank you.
Mm-hmm.
Thank you, Pixie, and thank you, Prof. Yuzaki, for the great talk. So we are going to move on to our next speaker. Our next speaker is Professor Shen Young. He's the Chief Scientist of the State Key Project on Aging and Organ Degeneration, National Science Foundation of China. So he's also the Director of Neurodegenerative Disorder Research Center, University of Science and Technology of China, and Institute on Aging and Brain Disorder, University Affiliated Hospital. Prof. Shen has received many awards, including Magnus Rudolph Dutch Young Scientist Award, Zenith Award in Alzheimer's disease, and Outstanding Contributor for Alzheimer Research from USA, as well as Changjiang Lecture Professor Award from China. Currently, he overviews aging research in a national-wide level, including aging mechanism, biomarkers, and intervention. Today, Prof. Shen will share his insights on translational aging research in China. So I'll pass the floor to you, Prof. Yong Shen.
Well, thank you very much. I can't share my slides right now. Okay, here. Okay, so really appreciate it. I'm very honored to be here and share our recent translational aging research in China. Let me introduce these programs in China, especially from the National Science Foundation of China. There's a national major projects on organ aging and degeneration studies. Okay, so this program, they call the Maximum of the Organ Aging and Degeneration on the major research projects from 2016 to 2024. So total of about 8 years. So now it's just finished the 4 years. So the midterm, which is the finish the review. And the government, the foundation invests 200 million direct cost Chinese yuan, and also 250 million yuan for total cost. We do have a scientific advisor committee. So right now I'm the chair for this committee to overview all of the aging research and degeneration related studies in China. The critical question we are trying to tackle in this program. For example, the first one is a basic characteristic. We're trying to identify specific molecule markers for aging and degeneration. When we say the biomarkers, for example, here, it's not only for neurodegenerative disorders, but also for markers could differentiate health aging versus the abnormal health, abnormal aging towards to degeneration conversion. And secondly, environmental factors. We're trying to dissect off internal and external environmental factors that drive health aging to degeneration. So the windows here is relatively small because we are not particular study neurodegenerative disorder. We're trying to focus our effort on the health agent converting to degeneration. The third one is regulates the most. For example, we're trying to identify is there any innovation approach or high technologies that can help and promote this aging research study. So today I'm going to have this the full aspect to outlines and also overviews of what's the entire programs, the objects. So I'll start first one, new mechanism of aging and neurodegeneration. As we all know, this year, actually, Shanghai and Jiao Tong University with the Science Journal proposed 125 most challenging frontiers scientific questions in the world. The two of them is related to our aging and degenerations. The question one is, can we stop or delay from aging? And second, can we cure neurodegeneration, for example, Alzheimer's disease? So as we all know, we compare in terms of the GDP or the country size-wise, the US, for example, as example, and the National Institute of the Health, or the one institute, for example, NIA. So they invest about 2 billion US dollars and every year in their budget for aging research, aging and related disorder. And the most powerful company, Google, for example, 1913, they invest 1.3 billion US dollars for aging and related disorders. So, and also the top journal, for example, Nature and Lancet, recently they launched out aging-related journals, for example, Nature Aging, and Lancet Health Longevity. So that's, again, illustrates aging research becomes a really hot topic in global frontier science. And also, back to China, for example, the WHO's report from last year, the projected population, the longevity is 76.4 years. However, the average, the projected the health longevity is only 68.7 years. It's about 8 years difference. The life with chronic disease, CD. So that about behind the developed countries. So in other words, the elderly in China suffers from CD early and also with a long time and poor life quality. And also everyone knows probably that in China now is the aging rate for the elderly population is really go really quickly. For example, last year, 2020, is 180 million the population above 65 years old. By 2030, it's 400 millions about 65 years old. So this is really the huge population. And interesting, you will see is the most of those elderly, for example, 70% of this elderly in China have at least 2 chronic diseases. For example, the cerebral vascular and heart part and cancer, etc. So in other words, so number of elderly suffering from chronic disease and disabilities in China is the number is shot, it's amazing, which has a huge medical and economic burdens on the family and society. So in our program, for example, when we talk about organ aging and degeneration, It's really the key factor for chronic diseases. Aging really result in the organ physiological function decline, for example, brain shrink, the liver problem, and vessels as well. And also as the aging increase, the chronic disease frequency goes up directly. It's really shooting up. So let me share with you our some of the achievements in the past 4 years. First, in the new mechanism of the aging and neurodegenerations. So in our programs, and the scientists in our program discovered the new genetic mechanism of the longevity genes in the health centenarians. Those that we identify, for example, the centenarians of 76 cases, and their children about 54 cases in the Hainan province. And then compare with their spouses, contemporaries with the children. And so after we did the RNA sequencing of the peripheral white blood cells, you can see we do find very interesting result. It's about the autophagy and the lysosome pathway gene. So these are the genes our scientists identified significantly upregulate in centenarians, and also could be partially passed on their offspring. So this research is very dramatically, and then the published genomic research, the cover page in 2020. So that indicates so possible, that possible we could use this signal transduction outphage, the lysosome pathway genes for the healthy aging populations. When we talk about genetics, we have to deal with folks of epigenetics. So our scientists in this program identify some critical genetic regulators for brain aging. So for example, they identified BAS2B and EHTN1. So we know normally when people getting old, their memories, cognition functions start decline. However, when the abnormal memory problem, so they developed towards the dementia. And so when our scientists identify these two genes, interesting, when the people getting the old, there's the two genes expressing significance increase, and also when the more cognitive impairments decline, and the more these two genes expression goes up. And then if we knock out these two genes individually, and then we could delay the aging and also stop and delay some of the new degenerative disorders. So this is the first time for the gene regulatory network for brain aging. It's a worldwide, and which provide new intervention targeted for delayed brain aging. So when this paper published in Nature, so it's really praised by all of the experts in the field. Another thing is when we talk about Alzheimer's, we have to look at the ApoE genes. Our scientists finds very interesting results here. As we know, ApoE genes is divided into three subtypes: ApoE 2, 3, and 4. Only ApoE4 is the biggest risk factor for Alzheimer, as we know. And also our scientists found this astrocytic ApoE could reprogram neuronal metabolism. So they could regulate this neuronal much small molecules metabolize, and eventually, for example, cholesterol and other molecules, eventually could affect learning and memory functions. So this paper just published Neuron this year. And also, the scientists actually, since our immunologists found very interesting, is we found that the NK cells resident in the dendrites in the neurogenetic niche of age brains. Interesting thing also, the NK cell, when this NK cell depletion can lead substantial improvement in neurogenesis and cognitive functions during the normal aging. So this findings really provides very helpful possible this potential therapeutic approach for neurodegenerative disorders. The next, I'm going to talk about other achievement, the hallmarks of aging and neurodegeneration. As we all know, so when we deal with the clinical problems, we do need a biomarker for early diagnosis. But when we study the normal aging process, we also need to have some biomarkers to differentiate when it starts aging. How's health aging differentiate from abnormal aging? So the people in our program study, for example, the young monkeys and old monkeys, and now compare the human tissue. So they study different organs, for example, retina, lung, heart, and blood vessel, ovary, skin, and blood cell as well. And they found very interesting. Not only in the traditional hallmarks of aging, for example, the senescent cell accumulation and stem cell exhausted, endothelial impairment, and also intracellular communication problem, for example, inflammation pathway disabled, and some of the dysfunction about subcellular organ, for example, mitochondrial and ER. But also the scientists in our program identify some key regulatory molecules, for example, interleukin-7, KLF6, ACE2, and FOXS3, etc. So these molecules, regulatory molecules, could differentiate or in the critical biomarker differentiate the aging process, which could provide this not only so-called diagnostic, but also therapeutic basis for delaying the aging. And one of the organ they discovered, for example, is the ovary. So the ovary they found is a really dramatic change, and they do share some of the common characteristic compares the human ovary tissues. So this study published this in Cell in the last year in the cover story, and also selected the top 10 scientific advanced life sciences of China award. Also, our scientists, by using 3D facial imaging analysis to predict heterogeneity of the human aging rate. For example, it's interesting, we're trying to introduce the concept of biological age. Is this the two persons in the left? You can see the two persons, they are at the same age, 40 years old, but they look really different. So obviously the left one is much younger, looks younger, and the right one looks a little bit senior. And so then what is this is the so-called biological age. So if we use, for example, biological age minus chronological age, that is the aging rate. So then how we can to evaluate the aging rate? So by this, the 3D facial imaging analysis based on 5,000 individuals. So, and then combined all of the multi-omics data, the metabolites, protein, etc. And then we find out the biomarkers and influence factors of aging. So they calculate it's a mathematical model to evaluate the aging rate. So this paper is published Nature Metabolism. And for Alzheimer's disease, for example, we also identified some of the biomarkers. One example is, which is recently published in the Alzheimer's dementia last year. So they found the sleep. The sleep is correlate the U shape, the converted U shape, U shapes with the Alzheimer biomarkers. So that indicates if you sleep over, it's more than, for example, 8 or 10 or even more hours, or you sleep less, maybe 3 or 4 hours, they both could potentially got Alzheimer. So our physicians told us then, the maintenance you sleep quality is critical to prevent Alzheimer or prevent dementia. Maybe 6 and a half hours, maybe the high quality sleep that can help prevent dementia. And also the people also found in our plasma blood, where they identify A beta. As we know, A beta is the pathological key molecules in the Alzheimer. And if we identify the antibody to the N-terminal of A beta molecules, so that level of this antibody of A beta is direct correlated of the cognitive decline. So that indicates we can monitor this antibody level to predict and provide the drug treatment or efficacy of the Alzheimer disease. So the next achievements we like to share with you is intervention strategy for aging and neurodegeneration. So our scientists in the program identifies the interesting idea. So they screen the molecules. They use Huntington disease, which is genetic neurodegenerative disorders. So mutant, for example, mutant Huntington's protein, we call the mutant HTT in general. When this protein is aggregate, it's become very toxic to the neuron, especially to the striatum area. So the stripe in the pathological wise, you will see the striatum atrophy. Then how are we going to improve or reduce these molecules? The scientists discovered screen the small molecules which can stick on, can bind this mutant proteins, and one side and other side of the molecules can also bind some degradation proteins. So it's become we call the MHTT LC3 linkers compound, and linked together, they can bring this whole complex into this protein degradation enzyme, degradation pathway. So that way can decrease mutant HTT's protein level and therefore can rescue Huntington disease phenotype in vivo. So this paper is published in Nature, and it's also bring the possibility of the targeting proteins for degradation by using autophage. Then they can provide a new entry protein for drug discovery. Another, the therapeutic approach we discover is we understand misfolded protein. It might the critical factors which affect in neurodegenerative disorders, but also not only misfolded proteins, the whole system of the inflammation and also the neuronal cell death in the brain. This is three. It's a very typical in the neurodegenerative disorder, for example, Alzheimer's disease. And we use the chemical genetics approach to screen the molecule. We screened 170,000 molecules, identify our target, which could inhibit replicate 1 kinase, and also same time can increase autophagy. So that way we might be able to inhibit inflammation and cell death. Actually, we did. We identify molecules we called APT1, which could inhibit. We can inhibit tau aggregation, and also in, for example, in the CA1 area in the PS19 mice, and also could reduce inflammation and also prevent the cell death in the tau Alzheimer models. So that way, the one molecule we can kill the 3 birds: misfolded proteins, inflammation, and the neuron cell death. So that way, we could potentially convert the clinical part for the treating those neurodegenerative disorders. So this molecule is under the clinical trial phase 2. Besides those molecular and cellular levels therapeutic approach, so we also want to see whether some we call the active healthy program, or we can also see caloric restriction reprogram, and then can single this cells transcription. That way we could maybe help the health agent and prevent some potential degenerations. For example, we give these animals food restriction, and interesting, we see after we give the single cell sequence, we identify the CR, the calorie restriction, really reverse the aging, disturbed the immune system, reduce the significant inflammation a lot. And also we identify quite a bit of new biomarkers, and we can see whether the aging could significantly delay. So this research is published in the Cell last year, 2020, and obtained the top 10 scientific advance of China world. So next achievements we like to share with you is we do have a really focus on developing new technology. And this technology including 2 parts. One is developing new animal models, and other is developing new technology to evaluate the aging research. So for example, we recently developed a new big animals models. As I just mentioned, for example, the Huntington disease, we saw the striatum area have a pathological change. We call the striatum atrophy caused by the mutant HTT. However, you will not see this pathological problem in small animal models like a mouse or rats Huntington disease model. So then it will be difficult to evaluate this either therapeutic effects and the drug efficacy. So we have to find the bigger animal model. Then the scientists in this program established we call the brain region selective neuropathological model in the Pike. So we knock in this Huntington disease gene into the pig, and we do see this striatum atrophy in the pig model. So this opened a door for us to, we are, for example, we are expanding this research to other type of neurodegenerative disorder, for example, Parkinson's disease, as well as Alzheimer's disease in the pig models. We also trying to set up some the monkey model. So, for example, in our program, we generate Hutchinson Gilford's Progeroid syndrome monkey models. We do see this monkey models is really shows sequence. All of the organs, for example, the change become the aging and degenerates we just previously introduced is from this type of the monkey models. So this models really give us a lot of tool to further evaluate from developmental stage to the aging process of the elderly stage. So so those are the two big animal models as we recently developed. Now I'm trying to introduce some of the new technology we developed in their program. One is a new technology we call the tracing protractor. So in other words, we're trying to identify any tracer or the we call the genetic approach tracer. We can monitor the single cell from developmental stage, a young age, for example, all the way to the adulthood and the elderly stage. So that way we can see how this aging changes the cellular level and see how they could interact subcellular organs. We also developed really a set of the probes. We call that really high performance probes. There was for example, there are NADH flex, NADH SONA, and NAD 5NAS, the probe. So that way we can monitor not only the cells, living cell, but also we could monitor this aging changes in the live animals by using this high performance probe. So this we call the real-time monitor of cellular metabolism in living cell and animals here. So we published this series papers in the past 3 or 4 years. And the last and also we developed the others that we call the single cell mass spectrum, the technologies. By using this single cell proteomics technology, we will be able to see the metabolism change at the single cells level. And also this technology allow us to see the single, for example, we can see the single small molecule metabolites in neurons, in the cortex, in the hippocampus. And scientists, by using this technology to identify a new, for example, glutamate biosynthetic pathway in the brain. So that's results published in Cells 2018. So, and also obtained the top 10 scientific advance in life science of China award. So this is what I can share with you today. We recently we believe make a lots of progress. And also, again, all of these studies is supported by the National Natural Science Foundation of China. And this major research program we again called mechanism of open aging and degenerative disorders. So we really appreciate the foundation's strong support, and they will again to support another 4 years. We are expecting there's some more translational and also more clinical trials been done after 4 years. And we really appreciate our scientific advisory committee make lots of critical evaluation and critical review and critical suggestion for these national projects. Appreciate. If any question, I'll be very happy to answer it here.
Thank you, Prof. Shen, for such an excellent talk and great snapshot into breakthrough aging research and also state-of-the-art technology or approaches from China. So really amazing that the country is investing a lot of money in not just basic research, but also the whole translational pathway. So that's really great. Every time we see investment into research, we all scientists get really happy and excited. So yeah, so just want to really go through some of the questions. Well, I'll just quickly kick off with one. So Prof. Shen, you mentioned about sleep quality. It's really important for this healthy aging. Could you perhaps comment on how we actually measure the sleep quality? Because you did mention when we sleep too long or too short, it's not that important. It's actually the sleep quality. So could you perhaps comment on how we could measure or assess, evaluate sleep quality?
Mm-hmm. So this part of the study has been performed by our neurologists in Shanghai Huashan Hospital. So they evaluate more than thousands of patients over there, and by using their EEG and also by measures the eye movement. So that we combine those two together. Of course, there was lots of the psychological battery test after that. They did a serious longitudinal study. So in other words, as we know, when the sleep, you can divide the deep sleep and versus non-deep sleep, right? And also by using the eye movement, they also can measure the fast eyeballs movement and slow one. So they use that to evaluate the sleep quality. Of course, in generally, so the common knowledge in medicine is the deep sleep more, for example, in general, is about 1/3 of your total sleep hours. That indicates your sleep quality is good. If if less than 1/3 of the time is sleep hours, and then your sleep quality is not great. So they have a details evaluation category to evaluate each individual patient to do so, and then they follow up this psychology battery test, and then of course they also have a biomarker to evaluate the cognition of each individuals. So combine together, so they draw this conclusion about this. So that paper is published in the last year's Alzheimer's dementia.
Thank you, that's great. So we've got a question from Dr. Shampa Ghosh. So his question is, do you think it would be nice to have common biological and application? So I would imagine this technology-based markers that can simplify in real-time scenario in human health issues. So for example, in Alzheimer's disease or metabolic disorder. And if yes, how could we go forward as a global level to decipher and collaborate on this?
Well, that's my goal too, as our whole program goal. So we would love to translate this technology, first of all, translate into the human. And then, of course, we would love to expand to the whole world. And in terms of those technology, for example, I'll take one example as a single cell proteomics, for example. This is developed in our university. So now is we focus on to analyze these single neuron metabolites. Now we're trying to move this technology into the blood cell. If we could develop this high throughput technology, and we can evaluate just not more than just one cell, maybe multiple cells at the same time and in the blood, then we could evaluate metabolites in the blood cells. Well, of course, we could do one single cell at a time in the blood, but that's one single cell does not represent the whole the blood cell population. So that's why we want to see the high throughput the screening to see more cells about that. And other, for example, the probe, for example, then we would like to. So right now we can see the animals. We would love to see whether we could change fluorescent, this probe fluorescent into the infrared, and then we can use apparatus to detect this probe. Then that way we could monitor this change during the, for example, disease progression, for example, of course, aging. So we love to share this technology with our colleagues in the field. When this pandemic is over, I would like to have everyone in our here to group together. We can exchange idea and bouncing this discussion, this our results, and maybe some brainstorm together. Maybe if not this year, maybe next year. That's our hope. So,
thank you, Prof. Shen. Just one last quick question. You've shown multiple or various pipeline or research pipeline that's coming up. So which one, or can you comment on which one are we expecting coming off the shelf in near future to help us stop or delay progression of neurodegeneration or aging related disorder?
So there were a couple. First of all, we have a bunch of the biomarkers to help us to evaluate this aging process and degenerate. So we can differentiate the normal health aging as well as degenerative disorders. So that one, a part of it already in our clinical cohorts there. We definitely can share with you the result after a couple of years. And also in terms of the intervention part, the two molecules I just shared with you, one we identified in the Huntington disease. So that one right now in the translational stage right now. We are in the, right now that part is in the phase 1, the study, and we have a milestone driven for that molecule. And meanwhile, we're expanding that technology into other type of the neurodegenerative disorders. And the other molecule, the APT1, for the Alzheimer inflammation, prevent neuron death, that one already phase 2. So that in the clinical trial in ALS first, not in the Alzheimer yet. So that's our hope to see we could finish the path. I think already getting close to finish phase 2. And if the result turns to the positive, we'll definitely go forward phase 3. And meanwhile, of course, we are expanding that one to clinical trial for Alzheimer's disease, since in small animals models for Alzheimer, result turned out very enthusiastic. Yes.
Thank you for your insight again, Prof. Shen, and thank you for your time. So for the interest of time, we will move on to the next speaker. There are a few more questions, I think, in the Q&A, so you can just answer it. So I'll pass the floor to Pixie. Thanks.
Thank you, Lin Kui. Hi, everyone. Thank you for staying with us. And here we welcome the 6 speakers, in fact, the last speaker of this second webinar horizon. She is Professor Julie Bernhardt from Australia. Professor Julie is currently the Deputy Director of Florey Institute. She has worked with stroke patients for over 30 years. She co-heads the stroke theme at the Florey Institute of Neuroscience and Mental Health Australia, leading a research team developing and testing intervention to improve stroke recovery. She is also the director for the National Health and Medical Research Council of Australia Centre of Research Excellence in Stroke Recovery, and also the world leader of multi-country rehabilitation trials. She leads 2 international stroke recovery and rehabilitation roundtables that set new research standards, culminating in development of a new International Stroke Recovery and Rehabilitation Alliance, ISRRA, in 2020. Prof. Julie is also a strong advocate for women in science. So due to her overlapping schedule with another event, so she has kindly pre-recorded her video to share with the attendees today. So I will pass it to the IBRO Secretariat to play the video, and attendees, you can actually also type in your question in the Q&A box that we will forward your questions to Prof. Julie. Thank you.
Hello, thank you for inviting me to give this talk about neurorehabilitation and the big picture. It's really lovely to be here, and it's a shame we still can't be there in person, but I very much look forward to that. So I'm actually going to focus in not on the process of rehabilitation, and I'll come back to that, but the idea of stroke recovery and at the different levels at which we think about recovery. So we think about recovery at the brain level, at what might be happening in an injured brain. We do research and think about what happens to the individual and all of the psychosocial consequences of stroke. And we also do think about the family and how their relationship is so important in the stroke recovery process. We also think about the environment in which rehabilitation or recovery can occur. And I think all of these are important aspects of our attention when we're thinking about recovery after stroke or brain injury. In terms of the big picture, we have a large number of trials of stroke rehabilitation type interventions, and they cover a spectrum of research and a spectrum of the disorders that we see after someone has a stroke. What you can see from this slide is the increasing number of published randomised trials and reports about stroke rehabilitation. And it really is a phenomenally large amount of research. But unfortunately, despite these very intensive and active research, we really are having a bit of a snail's pace in progress in recovery. And so we've been spending time wondering about whether this is due to suboptimal research designs, whether we're not working at the right time for repair of the brain, and also we've tended to work in isolation from each other by discipline and by country. And so these are barriers, I think, to progress in our field. But we have very big research questions that we want to solve, and there is this large amount of research output, which also means that we're spending a lot of money on research, and yet at the same time we struggle to get research funding for stroke rehabilitation and recovery. So I think there is this problem that we have, that without showing progress, we are really not favoured in terms of getting funding for the research that we want to do. Now, I do want to go to a few of the definitions that the Stroke Recovery and Rehabilitation Roundtables have spent time focusing on. And so when we talk about rehabilitation, we're trying to really limit that terminology to being the process of care. So the way that care is delivered. Interventions within rehabilitation are rehabilitation interventions. In terms of recovery, we want to think about the extent of return to pre-stroke state. And here we're particularly interested in both understanding more about spontaneous recovery, but also really getting some progress with behavioural restitution rather than compensation, because that's really what we're aiming for. We also want people to consider the timelines and become consistent in the language that they use for describing times after stroke. And this we would like to be linked to known biology of stroke recovery. So in our very first Stroke Recovery and Rehabilitation Roundtable, we put in a figure like this, which you can find in the 2017 paper, that really is trying to get people to have this consistent terminology around the timing. So hyperacute, acute, early subacute, late subacute, and chronic. Now, we also recognise that these are quite gross, but until we have something that is much closer to really understanding the time course of recovery and perhaps even individualising that information depending on someone's brain readiness for rehabilitation, then these are what we are liking people to use. Now, what we know is that there is probably a period earlier in a person's stroke recovery pathway where they are most likely to get gains when interventions are applied in that time point. And yet, sadly, a lot of the research that is happening in rehabilitation and recovery is occurring at a much later time point in this sort of chronic phase after stroke. Now, that's not to say that we should not be doing research in the chronic phase after stroke, but if we really want to make progress, we want to try and get some gains and show that we have some interventions that have impact for our patients. We know that brain repair and reorganisation is possible. And I think that this is one of the important concepts that we need to really hold on to and work towards understanding more how we can optimise these processes. Well, what will the breakthrough treatment look like? It's possible that it will be a combination. So it could be multimodal. It might involve a recovery promoting drug in combination with behavioural treatments. We just don't know, and we need to unpack that. But we also think that the place where people do their interventions or where they live has some bearing. So in the rest of this talk, I just wanted to tell you a few examples of some of the international collaboration efforts in stroke recovery and tell you a little bit about the idea behind them, why they started, and what they're trying to achieve. And while there are many examples of international collaborations, I'm just going to focus on these four, which is the work from the Stroke Recovery and Rehabilitation Roundtables, and also aphasia trialists, the Genetic Alliance, and I will tell you a little bit about the hospital redesign project that I'm involved in. There are other examples, the Secondary Prevention Network, and also the Sitting Time After Stroke Network. And I won't be talking about the Early Rehabilitation Trials Network that I lead, but I would like to acknowledge that I have some wonderful collaborators, particularly in Malaysia, who are involved in these projects. So I did mention I will talk about reimagining brain recovery environments as well. So these are the 4 areas that I'll quickly take you through. So for the reimagining brain recovery environments, this is our novel redesign project. It is an international collaboration, and we've created a living laboratory which involves multiple stakeholders from very different backgrounds, from government, healthcare, from industry, and that's architects and designers, from academia, and also from stroke survivors and their families. And what we're doing is looking at the evidence base that underpins recovery environments. And through a process of very fun and interesting workshops that we held last year, we've started to get a sense of what is really different and important that isn't in the environments we have so far, and how we might be able to design these into our new recovery environment. The end product that we hope is to optimise rehabilitation design through research. And you can find out more about that by contacting me later. The second group I want to acknowledge is the collaboration of aphasia trialists. Now, this group began in 2013, and they wanted to coordinate international aphasia research. And so through this networking process, they wanted to really build collaborative projects and start to put some standards in place for aphasia research. So their scope is aphasia assessment, diagnosis, prognosis, rehabilitation interventions, recovery and reintegration of people into society. This is truly an international collaboration and has been funded by the Tavistock Trust in the UK. Now, they've had more than 28 individual projects that have been part of their collaboration, with 25 publications. They've had 17 research exchanges involving 15 countries. They run training schools, writing workshops, and international aphasia research strategy development. And they're starting to work together to create collaborative grant submissions. So if you're an aphasia trialist or an aphasia interested person, I really encourage you to contact this group and get involved. They also developed some core outcome measures for aphasia studies, and this was published in 2019. The second group I want to talk about is the GISCOME group. So this is from the International Stroke Genetics Consortium, which has been running for some time. And this is a special group that has been formed to look at stroke outcome. And the idea behind this group is to investigate genetic variants that might influence ischemic and hemorrhagic stroke outcomes. And they really believe that understanding the biology of recovery involves delving into the genetics of stroke. So this group have just published in the International Journal of Stroke some standards of what they believe should be recorded measures that are included in any genetic studies if you want to study recovery and genetics. So I recommend that to you. The third idea, and this is one that I have had the privilege of leading from inception, is the Stroke Recovery and Rehabilitation Roundtables. So the SRRR. And the idea behind forming this collaborative group was to engage eminent stroke recovery minds to think about the gaps that we have in our research processes and in our research questions, and really start to interrogate how alignment and collaboration could improve our ability to actually get answers for our patients. The first one was held in Philadelphia in 2016. And we've written a number of papers from this, including moving rehabilitation research forward, but also thinking about translation of basic science into clinical science, so better alignment between basic and clinical groups of researchers, biomarkers of stroke recovery, intervention monitoring and development for rehabilitation interventions, the standardised measurement that we recommend for all clinical trials of motor recovery, and the agreed definitions and timelines. All of these papers are free online in the International Journal of Stroke, and you can also get reprints from NNR. So this was so successful that we decided to keep going and ran the second Stroke Recovery and Rehabilitation Roundtable in Montreal in 2018. So we really wanted to delve a bit more deeply into how our trials need to change if we're going to reduce negative or neutral findings and really start to get some exciting findings from our trials. We also recognised in the previous roundtable that we hadn't addressed cognition, and that needed some attention from experts to talk about definitions and consensus. We also wanted to look at how we should best translate evidence to rehab practice, and as well, how do we measure recovery? So motor control and improved quality of movement, not just compensation, which is what most of our scales measure. So these also have been published, these recommendations, and they're all available free in the International Journal of Stroke. So I'm going to spend just a minute talking about how our trials need to change, because we feel that this is a really important aspect of improving the research that happens in stroke rehabilitation and recovery. So this was our task force. We also had some additional people that we consulted as part of the process, so an extended advisory. And importantly, a number of these researchers are basic scientists. So Tom Carmichael, Dale Corbett, Numa Dancouz, and Randy Nudo and Teresa Jones. So why did we want basic scientists part of our trials team? And that's because we feel that trials in both preclinical and clinical science need to change. And we need to think about how we build these collaborations between basic and clinical scientists to improve stroke recovery. So we created a stroke recovery and rehabilitation roundtable trials development framework. And this is a step-by-step pathway that we're encouraging people to use as they consider going on with their trials and what they put in to their clinical trials or their basic science trials. It is a tool to critically examine key, what we've called knowledge units when you develop your treatment trials, and I'll come back to that in a minute. But ultimately what we want people to think about is, should we go ahead with this study? Do we have enough knowledge to do it and do it justice and have a great chance of it actually finding a good effect? Or should we not go yet into a large trial or to a formulated trial because we actually don't know the answers yet? So our knowledge units include examining how much intervention you might apply, what are the active ingredients within that intervention and how do you carefully describe it, who is the target group for your intervention, and when in the recovery pathway should it be applied. So there's a lot of detail in this paper and I really urge you to go and have a look at it. But we also are considering adjuvant treatments and whether they're paired with behavioural training, and you'll see some information in that too. Essentially, what we are hoping is that people carefully consider how they do their trials, and if there's not enough knowledge, then don't go ahead with large clinical trials, but instead think about alternate models to interrogate different specific questions and get that information before we go ahead and do large trials. So there is some information about some of the earlier phase trials that we're recommending, which include dose escalation trials and biomarker enriched trials. And you can read more about that in the paper. But what we really took away from running these 2 roundtables was that we wanted to bring more people together, and that roundtables by their nature, because they're small and focused and have task forces, are not broadly inclusive. And so the idea that we had was to create an international alliance around stroke recovery and rehabilitation. So we met in 2019, at the end of 2019, before the pandemic, and discussed a strategy for developing this international alliance. So ISRA was officially formed then, and in 2020 we had a virtual meeting that confirmed that we would like to go ahead and create this international alliance. Now, we've been spending quite a bit of time, this is sort of a strategy outline, working through our milestones over on the left-hand side of this slide, and we are looking for funding to help support the development of the alliance. It does need a bit of infrastructure, which we still don't have, but we've been busy still looking at developing flagship project ideas, and also to create ideas or criteria for aspirational centres of clinical excellence. We have successfully launched the alliance. So we've done a lot of work. We just still need probably to get some funding just to help consolidate. Now, this is open to anyone, and if you would like to join, you are very welcome to do so. So I'm going to conclude by saying that stroke recovery really is a challenging frontier, but it's also a very exciting frontier. And we're making very good progress in understanding the brain, understanding recovery processes, and developing and conducting our clinical trials. We're still not there, and we still don't have truly exciting progress that is impactful yet. But international collaboration is growing, and so I very much hope that in the coming years we will absolutely be making fantastic progress in this field. And I encourage anyone, especially the younger people in the audience who are interested in stroke, to get involved with any of these alliances. And really, you are so important for the next generation. So I just want to acknowledge the amazing people that I have had the privilege to work with over many years, including my team, including the Stroke Recovery and Rehabilitation Roundtables. And of course, we've had the wonderful Stroke Centre of Research Excellence that has been really formative in many of these ideas. So all of those individuals have been just terrific. So I just thought I'd leave this final slide for you to have a look at. And this is contact points for the stroke recovery, rehabilitation areas. And if you're interested in any of these contact details will help you find someone that you can talk to about getting involved. So I hope that big picture view has been useful for you and that you have a fantastic rest of your meeting. And thank you again for inviting me to give this talk.
Okay, so we have our last talk from our last speaker from Professor Julie Bernhardt. So I think we're just gonna wrap this webinar up. So for those who have any questions, feel free to fill up the Q&A boxes, and we will send it to Prof. Bernhardt, and she will personally respond to it. So do leave your email address so that she can reply your email. So just to wrap up today's webinar, First of all, I would like to thank IBRO and Fons, the organizing team, Tasia and Jonathan, for the support in organizing this webinar. It's a great pleasure to have Dr. Pixie as a co-chair for this webinar. Our sincere appreciation to all the 6 speakers for their time and also insights on the translational neuroscience and novel therapeutic for neurodegenerative diseases in the whole Asia-Pacific region. I'm sure that we all learned many novel and breakthrough research in this session. Clearly, it's important to focus on the discovery research as well as translating the finding to clinical applications. So one of the gold nuggets that we constantly hear in this talk or this session is we need to work collaboratively. We need to break down silos, and we not only work nationally, but also address this serious neuroscience research as a global moving forward together. So for those who are interested to rewatch the talk at later stage, this webinar will be uploaded in IBRO YouTube channel. Okay, finally, for all the participants, thank you for joining. Thank you for your time. For those who are interested, please click on the link provided and you will get e-certificate for participation. So thank you all for joining.
Thank you very much for your sharing. It's really appreciate your great effort. I learned a lot.
I agree.
Thank you all. Stay safe. See you again.
Bye-bye.
Thanks, Pixie. Thanks.
Bye.
Bye-bye.
I like your electrophysiology work. It's great.
Thank you very much. Your work also is very impressive. All your talks are fantastic.
Yeah, it's everyone. Cliff, I like your sheep model. Maybe I'll contact you.
And they are, I didn't mention, but the people are also have a Huntington's sheep model and developing an Alzheimer's sheep model also.
Oh, really? How are you going to evaluate the sheep cognition then?
I showed you one task anyway.
Yes, that's the global networking that we wish to achieve.
Yeah, I love. Maybe we'll invite you to give a speak in our future international neurodegeneration conference. So,
yeah.
Everyone.
I was supposed to go to one in Wuhan in 2020.
All right.
Okay, take care, everyone. Yeah, we have to sign off now and enjoy your evening.
This is a really great pleasure to see you.
Bye.
Save us.
Thank you all.